in this class, we'll be discussing about the genetics. Okay. Now, in this genetics, first itself I am telling you, see, what are the most important topics? In your exam, 100% there is no doubt there will be one question on the inheritance pattern, whether it is autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive pattern. They will give you one disorder and they will ask you what is the inheritance pattern, or they can ask something like, so which of the following diseases follows X-linked recessive pattern? All of the following X-linked recessive pattern except. So such kind of questions can come in your exam. Okay. Having said that, now let's begin with the genetics. See, what exactly is a gene? Okay, so what exactly is a gene? Now you can very clearly see here, this is a cell. Okay, here is a cell. Now inside the cell, we all know there is something called as a nucleus. Okay, there is something called as a nucleus. In the nucleus, what do we have? We are having chromosomes. We all have chromosomes, right? Humans have 46 chromosomes. Okay, 46 chromosomes, 23 from the father and the 23 from the mother. Okay, done. Now if you look at one chromosome, now what exactly this chromosome made up of? So if you unwind the chromosome, if you open the chromosome, what you are going to have is this material which is called as a chromatin. Okay, which is called as a chromatin. Now even if you open the chromatin, okay, so what exactly is this chromatin, sir? What exactly it's made up of? The chromatin is nothing but some nucleosomes and the DNA. So at the end of the day, the chromosomes are made up of this DNA. This is all the DNA, sir. Now in this DNA, what do you have? Sir, in this DNA, the sequence, see here, this is the one strand of the DNA, this is the other strand of the DNA. The DNA, what it consists of? It consists of nucleotides. You can very clearly see here, these are the nucleotides. Now, a series of nucleotides, or the sequence of these nucleotides, for example, from here till here, it will be one gene. Okay, so what exactly is a gene? So, genes are present on DNA. Genes are nothing but a sequence of nucleotides. Sequence of nucleotides or series of nucleotides is called as a gene. There are many genes. How many genes are there in the human body? How many genes are there? There are almost 20,000 plus genes. Okay, there are almost 20,000 plus genes. Now in these genes, okay, for example, you're having many genes. Do you think all these genes are going to be working? No, most of the genes are not expressed. So what are these exons, sir? MCQs in your exam. So what are these exons? Exons are those genes which are expressed in a simple way. Okay, in a simple way we can say something like this. So those genes which are expressed, exon for expression. So those genes which are expressed. Okay, so those genes are going to be expressed in the sense they will undergo transcription, translation, protein synthesis. Okay, so exons are those genes which will undergo transcription, translation process followed by protein synthesis. Okay, so introns. So what exactly are the introns? Introns are those genes which are not expressed. They are not expressed. So they are present in our genome. We have exons and introns in our DNA, but exons are going to be expressed and introns are those genes which are not expressed. Okay. Now, what do you think? Which will be more? Exons or introns? What will be more? Introns. Okay, introns are more in number. Okay, almost 98% are the introns. Okay, almost 98%, 98% of our genes are not expressed. Okay, now after this, let's discuss about the locus. See, first we are all starting with the basics. Okay, we are all starting with the basics from the chromosome level, from the chromosome, DNA, nucleus level. We are going one by one. Okay, what is a gene? How many number of genes are there? Where exactly the genes are present? Okay, now what is a locus? Locus, in a simple way, is a location of a gene. The location of a gene is called as a locus. Now, if you ask me, so where are you staying? I have to give you my address, right? See, this is the place where I am living in this country, India. I am living in so and so state, in so and so district, in so and so town, in so and so street. Okay, so that's the exact precise location. Okay. Now, even if you want to know where exactly is the CFTR gene present, now I have to give you pinpoint location. So this is the chromosome on which gene is located. So that location is called as a locus. Locus means the location of the genes on the chromosome. Okay. For example, you just look here. I'm giving you the example of CFTR gene. Okay. Guys, can you tell me where exactly is this CFTR gene is mutated, in which condition? There is this one condition where the CFTR gene is mutated. Okay, that's the cystic fibrosis. Now, look here. The gene is CFTR gene. Okay. Now, where is the CFTR gene located? See, the location of CFTR gene is 7q31.2. So, 7q31.2. Sir, what exactly is this 7q31.2? First, this letter number 7, it is denoting. Okay, this letter number 7, what exactly is this denoting? Letter number 7 means here 7th chromosome. On chromosome number 7, okay, first letter is chromosome. Okay, on chromosome number 7. Sir, what is this Q if you ask me? See, each chromosome have two arms, P arm and a Q arm. Okay, P arm and Q arm. P arm is the short arm and long arm is called as the Q arm. Even in this chromosome, you look here, the short arm is called as a P arm and the long arm is called as a Q arm. Okay, so the CFTR gene is present on chromosome number 7 on long arm. Okay, sir. Then where? See, this is all the long arm. Okay, now where, sir? Here. See, this 31.1. Okay, what is this 3 stands for? 3 stands for this region. Okay, region, sir. In the region number 3, see, 3 stands for what? 3 stands for region. And I, what is this? Sorry, not I, 1. 31, right? So, what is this 1 stands for, sir? 1 stands for band 1. In this region, this is the first band. In the band 1 and subband 2. So, this is the exact location of CFTR gene. So, now tell me, what is this 7q31.2? 7q31.2 means first letter stands for, first number stands for chromosome, PRQ arm, next region, band, subband. So, this is the exact place where the genes are located. Okay, they can ask you, they might give you, for example, in there is a syndrome called as DiGeorge syndrome. DiGeorge syndrome. Okay, DiGeorge syndrome. There is a famous question. There is a deletion of chromosome number genes in 22q11.2. Okay, so there is deletion of genes from chromosome number 22q11.2. Now you know, sir, what is this 22q? 22 is the chromosome number, q is the arm, 1 is the region, 1 is the band and 2 is the subband. Okay, so this is the exact location, guys. Now after this, what else you should know? Okay, done. So locus, now you know it. So what exactly is an allele? So what exactly is allele, sir? Alleles are the alternate forms of the gene. Okay, alleles, they are the alternate forms of a gene. Now you can ask me, sir, these are like little confusing words like alternate forms of a gene, because students have a very limited knowledge, especially in their first years. Okay, and second year, they have a very limited knowledge about the genes, chromosomes, alleles, okay, genotype, phenotype, these things. So what exactly is an allele? Hope already you have studied in your first year biochemistry. Alleles are the alternate forms of a gene, which means, dekho, you are having one gene. Okay, you are having a gene, sir. What is a gene? You already know it. It's a sequence of nucleotides. For example, imagine this is a gene. On the DNA, that particular segment is coding for a gene. Okay. Now, what it is coding for, sir? It's coding for the eye color. It's coding for the eye color. So, gene is one gene only. But, see, what are the different alleles? What are the different eye colors? Okay, the phenotype can be blue eyes, green eyes, grey eyes, okay, dark eyes. So, these blue colors, the expression, the expressing thing, these are the different variables. You are understanding? So, now you can have a one gene. Yes, gene is one, but these two are the alleles. You know it, black eyes, blue eyes. Not only just that, for example, you look here, this is, imagine, this is a gene. So, one gene is having how many alleles? See, there can be many number of alleles. Most of the students think there are only 2 alleles, sir. No, there are not just 2 alleles. There can be many alleles. Okay. For example, you can have the gene is one gene, the gene for eye color. This is a eye color gene. Okay. Now, this eye color gene, it may be coding for, maybe coding for dark color or blue color, green color or grey color eyes. Okay. Variations. Next, for example, here is a gene for hair color. It can code for different color hairs. Okay. So, a gene is having multiple alleles different variables, alternate forms is called as a allele. For example, let me tell you. See, gene. First, I will give you a gene. What is that gene for? You should tell me the alleles. Okay, you should tell me the different forms. Now, height gene. Gene for height. Height of the person. Now, can you tell me what are the alleles possible? What are the basic, okay? What are the alleles which you know? Sir, if height is a gene, how many alleles it can have? See, one thing is it can code for tall height or it can code for short height. Possible, tall and short. Simple. Okay. So, if for example, let me give you one more example. If gene is for skin color. If gene is okay for the skin color, what are the different alleles possible for the skin color? White color skin or black color skin or dark color skin, brown color skin, there are different colors. So these colors, these variables are called as the alleles. Okay, now we understand what is the difference between the gene and alleles. Gene is what it is coding for. Alleles may be different. Okay, different forms. After that, now the next term which you should know for your basic understanding at this level is homozygous and heterozygous. Later you will understand the patient is homozygous for so and so. The patient is a heterozygous for so and so gene. So what exactly is this homozygous and heterozygous? Okay, let me give you some example. Imagine my alleles, for example, look in me. For a height gene is height. Now for this height gene, how many alleles are possible? 2 alleles are possible. What are they? Tall and short. Okay, tall and short. Okay, broadly, just generally broadly classifying a tall, short. I'm just leaving the intermediate like an average and just taking it out. So tall and short. So for tall, I'm just representing it as capital T. Capital T is the allele for tallness. For shortness, let's take it as a small t. Okay, small t. So capital T is the gene for tallness. Okay, tall. And small t is the gene for shortness. So what is homozygous? Imagine in me. See, if there is a person who is 6 feet tall, okay, now I am telling you, he is having capital T and capital T. What does this mean by capital T, sir? Capital T? It means, dekho, now this individual, imagine, see, there is this guy, okay, who is 6 feet tall. Now, tell me, he is having this gene, sir, height gene. How many copies of this height gene can be present in him? 2 copies, one from the mother and one from the father. Okay, one from the mother and one from the father. Excuse me. One from the mother and one from the father. See, both from the mother as well as from the father, now he is receiving both the genes, sorry, I beg your pardon. Both the genes are coding for tallness. So he is homozygous. Sir. Why I am telling it's homozygous? Because both are capital T. Now for example, if I am the person, okay, who have received one capital D gene from the father, which is coding for the tallness, and one small t from the mother. Now the genetic makeup will be capital T, small t, capital T, small t. Now is this person homozygous or heterozygous? This is heterozygous. Why? Because from the father capital T is coming, from the mother small t is coming. See, the genetic makeup, simple, what is homozygous and what is heterozygous? In the genetic makeup, if you have same type of alleles, then it is called as a homozygous. If you have 2 different types of alleles, one capital T, one small t, then it is called as heterozygous. Now I will give you one example. Can you tell me? Imagine there is this one person, okay, who is having his genetic makeup something like this. Small t. Is he homozygous or heterozygous? What do you think? Is he homozygous or heterozygous? Small t. This is definitely homozygous. Which means, what exactly this mean by? He got two alleles, one from the father and one from the mother. Both are coding for short only. Okay, so this patient is going to be definitely short. Okay. Next. After this, what else you should know? You should know what is this dominant allele, sir, and what is this recessive allele. A dominant allele, what exactly is this? And this recessive allele, what exactly is this? Okay. Look, dominant alleles, for example, now I am the person, imagine I am the person who is receiving capital T from the father and small t from the mother. Okay, capital T is coding for tall height and small t is coding for short height. Okay, now in both of them, who is going to be expressed? See, this capital T, it's going to be expressed. Okay, it's a capital T, it's going to be expressed. Out of these two, see, one is coming from the father and other is coming from the mother, but still only one gene is going to be expressed. Okay, they will decide. Okay, out of these two, I am telling you, see, this capital T is a dominant gene because it is expressing. So dominant alleles, out of these two alleles, the one which is expressed, the one which is going to undergo transcription, translation, final expression is called as a dominant allele. Okay, so recessive alleles are not expressed usually when in a heterozygous state. Okay, now देखो, recessive allele, what exactly is a recessive allele? Usually they do not express. They do not express. But when they will express in homozygous state. Okay, they will express in homozygous state. So what exactly is this, sir? Let me give you one example. Look here, small t, small t. Okay, small t, small t. So what exactly is this small t, small t? Sir, small t, it's a recessive allele, right? It's a recessive allele. This small t is also a recessive allele. Now they will express. Now they will express. But in this condition, whenever there is a capital T, that is a dominant allele, now it's not going to be expressed. So now tell me, in this condition, dekho, now in this condition, the patient is going to be tall or the patient is going to be short? First of all, you know that's a heterozygous state. I am the person who is having one capital T allele, one small t allele, one from the father, one from the mother. So now in this condition, tell me, am I going to be tall or am I going to be short? This patient is going to be absolutely tall. Why? Because the recessive allele, yes, the recessive allele is not going to be expressed. Now, let's see some examples. Okay, let's see some examples. Now, this is, for example, A and B. Now, here we are having a, for example, male. This is a female, male and female, father and the mother. Now, father is having brown eyes and mother is also having brown eyes, brown eyes, brown eyes. Now, father is having, everyone will have two alleles. Okay, father is also having two alleles. One capital B, one small b. Mother is also having one capital B, one small b. Okay, now right. Now, tell me. So, father and mother, both are homozygous or heterozygous? Both are heterozygous. See, both father and mother, both are heterozygous in their genes. Now, whenever they have children, whenever they have some children, now combinations, how can it can be possible? Capital B, capital B. One capital B, one small b. Okay. Now, one small b, one capital B. And see, dekho, there is also a possibility that from the mother, the small b is coming. From the mother, the small b is coming, and from father, the small b is coming. And now see, whenever the recessive genes, dekho, whenever the recessive alleles, whenever they are in homozygous state, only then they are going to be expressed. But in all the other conditions, look here, capital B, that is homozygous, but both the alleles are dominant. So, definitely brown eyes. Here also see, this is heterozygous state. Now, this child is having one capital B allele, one small b allele, but he is having one dominant allele. So, because of this B, capital B, the child is going to have dark eyes. Okay. Now, here also same thing. Okay. I hope you can understand this. Same. Look here. Now, only question which I want you to know is, tell me, father is homozygous or heterozygous? The father is heterozygous, but mother is homozygous for the recessive alleles. Father is heterozygous, one dominant allele, one recessive allele. Now, in this condition, say, what happened? Dekho, say BB means when recessive alleles are in homozygous state, now they are expressed blue eyes. Okay, so this is the concept which I want you to know. Now, after this, The next topic which I want you to know for your exams is the codominance. So far you have said that whenever there are two alleles, one dominant allele, one recessive allele, only dominant allele is going to be expressed. The recessive allele is not expressed. Okay. But when you have two dominant alleles, okay, when the two alleles are dominant, what happens? Who is going to be expressed? Both will be expressed. Okay, when both the alleles are dominant, both will be expressed. So this is called as a codominance. Okay, combination. Okay, codominance. Both are going to be expressed. Now, look here. What is codominance? Both dominant alleles are expressed. Both the dominant alleles are expressed. Example is, this is the one important for your exam. ABO blood grouping and HLA genes. See, this ABO blood grouping and HLA genes, they follows this codominance pattern. Okay. Now, in this table, if you look, see, these are the alleles from the father. These are the alleles from the mother. See, for blood grouping, how many alleles are there? First, you need to understand. For tallness, not for tallness, for height, how many alleles are there? There are two alleles. That is capital T, small t, right? In the same way, for blood grouping, there are three alleles present, sir. Three alleles present. What are these three alleles? A, B, O. Okay, and even in the father, there is A, B, O. Now, when this mother and father, when they give rise to children, okay, when they give rise to children, what are the combinations possible? Once look here, sir, this A and A, when the father is A and mother is A, okay, what can be possible, sir? Now, the children will be definitely A. Okay, the children will be definitely A. Okay, now, father is A blood group, mother is B blood group. Father is A and mother is A. So now see, what I want to put into your mind is, look here, this A and B alleles, A allele, B allele, both are dominant alleles. Both are dominant alleles. As both are dominant alleles, in the baby C, they are having AB blood grouping. It's not A or it's not B, it's both AB blood grouping. Okay. Now, for example, A and father is A and mother is O. Okay, father is A and mother is O. Now, O is always recessive. Okay, O is recessive. A is dominant, B is dominant, O is recessive. Now, in this condition, now what the baby is going to be? Sir, you know, only dominant allele express. Recessive alleles do not express. So, when this father and mother, when they have this child, father is A, mother is O, the baby is going to be definitely A. Okay, now this baby's genetic makeup is A-O, A from the father, O from the mother. Only A is going to be expressed, O is not going to be expressed, A blood group. Okay, simply you can follow the table. Now, only point which I want you to know is, whenever there are two alleles which both are dominant, both will be expressed. This is seen in conditions of ABO blood grouping as well as HLA genes. Answer. Okay. Now, after this, next topic which we have to discuss is the genotype. So, what is genotype and what is phenotype? See, genotype and phenotype, most of the students confuses here. Genotype, let me tell you, first of all, look here. Genotype is the organism's genetic makeup. Okay, the organism's genetic makeup. In a simple way, we can write here, genetic makeup of an organism. Okay, genetic makeup of organism. You will understand better with the examples. Now, what is a phenotype? Phenotype is what you can see, what is getting expressed. Okay, how are the genes? Is the genotype. Okay, what is the composition of the genes? Like, you know, let me give you, for example, see, genotype versus phenotype. The genotype means, genotype of an organism is defined as actual complete genetic makeup of an organism. It refers to what? It refers to the pair of alleles, whether it is capital T, small t, or one capital T, one small t. Okay, whether they are homozygous, heterozygous, about the genetic makeup, how the genes are and how the alleles are. If you are talking at the level of alleles, then it is called as a genotype. Okay, now for example, see, now I am saying I am having one capital T allele, one small t allele. See, this is the genotype. We are talking about the alleles, like, you know, how the alleles are. And what is a phenotype? Now, because of as I am having one dominant allele and a small allele, what is the phenotype? The result, the expressed one that you can see from outside. It is tallness. This tallness is the phenotype. Now, for example, See, this is the genotype. I am homozygous for recessive allele, small t, small t. Then what is the phenotype? The phenotype is short. The phenotype is short. So this is the genotype. The genetic makeup is called as the genotype. What you can see from outside is called as the phenotype. Okay, the expressed things. Now, let's once go through this. Genotype of an organism is defined as complete genetic makeup of an organism. It also refers, look, it also refers to a pair of alleles inherited in an individual for a particular gene. That's a genotype. See, at the level of genetics, at the level of genes. Then what is phenotype? Okay, whether the type of blood, AB blood grouping, the hair color, the eye color, the skin color, the height of the person, what is getting expressed. Phenotype refers to an individual's observable traits such as height, eye color, blood type. Okay, that's what. Okay, so phenotype is determined by, tell me, phenotype, it is determined by the genotype. It's the genes that determine the phenotype. It's the genes that determines what is getting expressed. Okay. So even here in your class 11th and 12th you would have studied. See here BB. See, this is, you can definitely say this is homozygous, and capital means dominant. See, this is the genotype. Because of this genotype, there are purple colors. Purple color flowers you would have studied in your, just for easy understanding, I am just explaining you this. In class 11th and 12th you have studied the Grigor Mendel's experiments on pea plants. Okay. Now, small b means homozygous recessive. Whenever there is homozygous recessive, what will be the phenotype? The phenotype will be white colors. This is the phenotype. This is the genotype. Okay, done. Now, after this, let's explain you about the mutations. See, there are enzymes in our body. You cannot see the enzymes, right? Okay, there are proteins in the body. You cannot see the proteins. So, it's not just what is expressed to the eye, how the genes are getting expressed and what is the resultant product. You cannot see an enzyme or you cannot see a protein. Okay, but they are also getting expressed. So, phenotype is what is getting expressed, what type of protein getting expressed. Okay, but in a general understanding you can say what is the appearance. Yes, that's correct, easy understanding. Okay, now what are the mutations? Any idea? Permanent change in DNA material. A sudden permanent change in a DNA material is called as a mutation. A permanent sudden DNA change. What might be the reason? I might be exposed to ionized radiation, X-rays, or I might be exposed to some in a nuclear area. There is this area where the nuclear explosion have happened. Now, if I am the person who is in that area, because of the extreme radiation, that radiation can cause damage to my DNA material. So, damage to the DNA material, which is permanent, is called as a mutation, irreversible in nature, sir. Now, for your exam, what you should know is point mutation. How many types of mutations are there? There are two types of mutations, point mutation as well as frame shift mutation. Point mutation and frame shift mutation. So first, what exactly are these point mutations? Okay, point mutations, what exactly are they? Permanent heritable change, which means these changes, they can also get inherited to the offsprings. Okay, see, let me tell you point mutation means. Look here, this is normal gene. So DNA, in the DNA, here is a normal gene present. Okay, now in this normal gene, everything is okay. You can very clearly see here are the nucleotides, thymine, adenine, cytosine, thymine, guanine, cytosine, adenine. The sequence of the nucleotides are there. Okay, good. Now what happened is point mutation. Now, see, in this area, this T is replaced by C. Okay, the nucleotide, one nucleotide is replaced. Now, instead of T here, now it is replaced with C. So, it's just that one particular point, right? Not the entire chromosome is lost. The entire chromosome is not lost. Okay, only in that particular point, there is replacement of one nucleotide by other nucleotide. So, it is called as a point mutation. Okay, so what happens if there is a point mutation? Okay, let me tell you what happens if there is a point mutation. Three possibilities are there. Okay, if there is a point mutation, there are three possibilities. Before going to the possibilities, let me tell you an example. Look here, the example is sickle cell anemia. Yes. Okay, sickle cell anemia, sir. Normally, okay, wild type means normal. Okay, normal hemoglobin. This is the gene which is coding for the normal hemoglobin. Now look here, there is CTC. Sir, I am right now talking at the level of DNA. I am right now talking at the level of DNA, sir. The genes are present in DNA. Now what happened is, see, this is normal hemoglobin, CTC. Whenever there is CTC, first transcription will happen, right? First transcription will happen. So from this DNA, who is going to be synthesized? mRNA. mRNA will be synthesized. Dekho, this is the mRNA. Now, whenever here on DNA, whenever there is CTC present, now on mRNA, who will be present? Sir, C in the place of C, see G, A, G. Okay, wherever there is cytosine, on the opposite end, guanine will be present. Wherever there is thymine, adenine, and wherever there is cytosine, there will be again guanine. Okay, this is something normal. Okay, on mRNA, what you are having? GAG. See, this GAG, this is a trinucleotide. Okay, this is a codon. This GAG is the codon, which is normal. Now, this GAG codon is coding for what? It is coding for glutamic acid. So, in the hemoglobin, there is glutamic acid present. Okay. Now, what happened in sickle cell anemia? Point mutation. What is this point mutation? See, in the place of T, who came? A came. In the place of T, the thymine is replaced by adenine. Now, because of that, whenever there is a change in DNA, automatically there will be change in mRNA also. So, in the place of A, okay, see, whenever there is A, who will sit? Uracil will come and sit here, uracil. Okay, why? Because adenine, wherever there is adenine, the opposite, the complementary, you will be having uracil. Okay, you will be having uracil in the mRNA. Okay, so instead of A here, you are having now uracil. Now what? Sir, now this codon, this GUG, this codon, sir, this trinucleotide. Now this trinucleotide, this codon is going to code for valine. So in the place of glutamic acid, okay, the glutamic acid, now it is replaced by valine. So this is normal hemoglobin. So can you call this as a normal hemoglobin? No, this is not normal hemoglobin. So this is sickle cell hemoglobin. So now you have to tell me, sir, in sickle cell hemoglobin, what happened? Now the glutamic acid, okay, the glutamic acid is replaced by valine. The glutamic acid is replaced by valine because of a point mutation in the DNA. Whenever there is a point mutation in the DNA, there will be a change in the mRNA. Whenever there is a change in mRNA codon, automatically it codes for a different amino acid. That will cause change in the proteins. Now hemoglobin is getting affected here. This is a sickle cell hemoglobin. Okay. So done, sir. After this, what else you should know? See, because of these point mutations, what are the possibilities? The three possibilities are there. Okay, point mutations. Can lead to. Okay, point mutations are of three types. Okay, I should say point mutations are of three types. Before discussing they can lead to, point mutations are of three types. What are they? First type are called as silent. Silent mutation. Second one, nonsense. Oh, before telling you nonsense, first let me write missense. Missense mutation. And third type are called as a nonsense. So now tell me, what exactly are these? Sir, point mutations are of three types. Point mutations by themselves, they are three types, sir. What are they? Silent mutations, missense mutations, and nonsense mutations. Okay. So first of all, let's discuss about what is a silent mutation. Okay. So what is a silent mutation? Look in this example. Okay. Look in this example. Sir, normally it should be GAA on mRNA. Okay, on mRNA, it should be, the codon should be GAA. If the codon is GAA, then it codes for which amino acid? Glutamic acid. It's coding for glutamic acid. Now what happened is point mutation, one small point mutation in the DNA. So that in the place of adenine, now who came? Guanine came. It was supposed to be GAA. Now in the place of this A, now who came? GAG. Now, but still you should understand certain things. one amino acid, देखो, one amino acid, same, glutamic acid, one amino acid is coded by many codons. There are many codons, okay. Like for example here, in this case, GAA is coding for glutamic acid, even GAG, that codon, it will also code for glutamic acid only, okay. One thing, never ever forget, one codon codes for one amino acid, that's it. One codon codes for one amino acid, but one amino acid have multiple codons, okay, done sir. But here, because of the mutation, A is replaced with G, A is replaced with G, but still the amino acid is still the same, glutamic acid only. So if there is any problem now, will any problem occurs now? So there is no problem. So this type of mutations are called as a silent mutation. Mutation occurred, point mutation occurred, but still there is no issue because it's the same amino acid. So this type of mutations are called as a silent mutation. Next sir, next. Dekho, this is the codon GAA. These three nucleotides are called as one codon. GAA, this is called as a one codon. Now this GAA, it is coding for what? It is coding for glutamic acid. It is coding for the glutamic acid. Okay, well and good. But some point mutation occurred in the DNA. So in the RNA, see, it was supposed to be GAA. Okay, the codon should be GAA. But now what is happening in the RNA? Now, in the place of G, U is coming. Okay, in the place of G, U is coming, uracil is coming. In the place of guanine, uracil is coming. So whenever you are having UAA, this is called as a stop codon. So immediately the protein synthesis stops. Translation stops. So these are codons. Okay, UAA, these are the codons which will stop the process of translation. Immediately translation will stop, sir. So this type of mutation where a stop codons are getting produced because of the point mutation, now it's a stop codon. So they are called as a nonsense mutations. So nonsense mutation means there is, because of the mutation, now you are end up with a stop codon. Now in your exam definitely they will ask you, what are the stop codons? Which codons are responsible for stopping the process of translation? Okay, what are they? UAA, UAG and UGA. Okay, see these three codons, okay, in the entire world there are these three codons which will stop whenever you are having this codon in the mRNA, whenever you are having this codon on the mRNA, see, translation is happening with the help of the ribosome is doing translation. Proteins are synthesizing. Now, whenever this ribosomes, whenever they comes to this UAG, UAA or UGA, immediately protein synthesis will stop, sir. Okay, so this is called as nonsense mutation. Silent mutation I have explained. Point mutation leading to what? No problem. There is no problem because same amino acid is produced. Next, nonsense mutations, stop codons. And what is a missense mutation? What is a missense mutation? Now, see, it was supposed to be GAA. Okay, it was supposed to be GAA. It is coding for glutamic acid. Now, in the place of adenine, cytosine is coming and sitting in the mRNA. Now, it's a different codon, totally different type of codon. It's not GAA, it's now GAC. Now, whenever there is GAC, it codes for what? It codes for a different amino acid. Which amino acid? Aspartic acid. So, instead of glutamic acid in the protein, now there is aspartic acid. So now this is not a normal protein. This is definitely a defective protein. So this type of mutations are called as a missense. Okay, missense mutations. Okay. Now you should tell me this example of sickle cell anemia. It is point mutation, okay, but is it nonsense? Is it silence? Or is it missense mutation? Now tell me. Guys, tell me. The sickle cell anemia, there is replacement of glutamic acid by valine. So it is an example of which type of point mutation? Missense, nonsense or silence? Okay, so it's an example of missense mutation. Done, sir, done. Okay, so now just for me, tell me how many types of point mutations are there? There are three types of point mutation. What are they? Missense, nonsense and missense. Missense, nonsense and silence. Okay, done. After this, let me discuss about the frame shift mutation. So frame shift mutation. So what exactly are these frame shift mutations? Now look here. Now imagine there is this mRNA cell. Okay, transcription completed. Genes are perfectly normal. Okay, for example. Now Okay, let me put it this way. So now, see, imagine this is a DNA. Okay, here, imagine here is a DNA. Okay, sir. Now, in the DNA, what do you have? You are having amino acids. Sorry, not amino acids, nucleotides. Okay, in the DNA there are nucleotides. See, imagine these are all the nucleotides. Okay, nucleotides sitting like adenine, guanine, cytosine, thymine. Okay, adenine, guanine, cytosine, thymine, same. This is the DNA, imagine, DNA. Now, from the DNA, who will come? So, from the DNA, mRNA is going to be produced. Okay, mRNA is going to be produced. Now, in the mRNA, what do you have, sir? The complementary things, right? Complementary things. Now, here also same. Now, on the mRNA, now here also you are having the nucleotides. Okay, these are all the nucleotides coming. Now, what happened is, normally, we will take 3 nucleotides, right? 3 nucleotides is considered as one codon. 3 nucleotides is considered as one codon. Okay, this is something normal. 3 nucleotides is considered as one codon. Now, what happened? Sir, one of the nucleotide, okay, here, now at the level of DNA, I am talking at the level of DNA. Dekho. Now, one nucleotide you are adding here. Now, there is addition of a nucleotide. Now, whenever one nucleotide is added, same in mRNA also, one nucleotide will be added, no doubt. Okay, now, dekho, what is happening? Now, one nucleotide is being added here. New nucleotide. Now, what happens? Now, these 3 will become the codon. Now, these 3 will become the codon. So, entire codons, there is a shift. Okay, now entire protein is going to be affected. So, just because insertion of one nucleotide extra, and the same way, deletion also, addition or deletion can completely shift the frame. Okay, can completely shift the frame. So, let me give you a better example. Look here. Now, I am going to discuss about the frame shift mutation. Clearly see here, frame shift mutation. Okay. Now, here are the codons. See, this is the mRNA. Now, in the mRNA, adenine, uracil, guanine, adenine, guanine, uracil. Now, you know it. 3 nucleotides are considered as one codon. See, these 3, AUG is coding for methionine. AAG is coding for lysine. UUU is coding for phenylalanine. Okay, amino acids. Okay, good. But what is happening? Okay, what is happening? See, when a base pair is inserted, now insertion I have explained you, one base pair, one nucleotide, it is inserted, sir. Okay, now see, extra U, it should be what? It should be AUG. Now, what happened after AUG? See, after AUG, one base pair is inserted. Now, first is AUG and second one is AAG. Now, look here, this extra U is added. Now, what had become? Now, first, okay, no problem. See, AUG is coding for methionine, but because of insertion of this extra U is coming here. Now, these 3 will become the codon UAA. Now, you know it. UAA is what? UAA is a stop codon. So, immediately what is going to happen? Translation is going to be stopped. Okay? Translation is going to be stopped, sir. Now, what I am trying to put into your mind is, if you add a base pair or if you delete a base pair, what might happen? The entire reading frame is going to shift. See, this is called as a frame shift mutation. Now, you can look here very clearly. Okay, you can look here. Sir, base pair deletion. If you delete one base pair. Okay, if you delete one base pair. Now, see what happens. Normally, what it should be? Just look here. It should be AUG. First 3 should be AUG, AAG. After that, UUU. Look here. It should be UUU. One U is removed. For example, if I remove one U, that's what I am doing, sir. One U is removed. If you remove one U, first AUG is coding for methionine. Okay, sir. AAG is coding for lysine. Okay, but it should be UUU. You have deleted one U because of some mutation. It's gone, sir. Because of some radiation or something, it's gone. Because of some chemical, because of some toxin, it's gone. Okay. Now, this will become the codon. UUG will become the codon. Now, UUG code for different amino acids. Now, it is coding for leucine. It was supposed to be phenylalanine here. It was supposed to be phenylalanine. Now, in the place of phenylalanine, leucine is coming. Frame shifted, right? Now, see, it was supposed to be, after UUU, it was supposed to be GGC. GGC should code for glycine. Now, what is happening here? Because of the shift, now it became GCU. Now, this GCU is code for total different amino acid. Entire protein structure is going to be affected, sir. Okay. So, what I am trying to put into your mind is only one thing. Point mutations are of 3 types. What are the 3 types of point mutations? The 3 types of point mutations are silent mutation, missense mutation, and nonsense mutation, and frame shift mutations. Okay, the second type of mutations which I want to discuss here are the frame shift mutations. See, these frame shift mutations, the frame shift mutations are because of either addition of a base pair or a deletion of a base pair. The entire reading frame will be shifted, which will cause damage to the protein, which will cause entirely different amino acids going to be present in the protein, which will cause the damage to the proteins. Okay, entire protein will be affected, sir. Defective protein will be formed. So done. Now after this, let's discuss about genetic disorders. Okay, now let's discuss about some genetic disorders. Now, the genetic disorders are mainly classified into 4 types. What are they? Mendelian, those genetic disorders which are following the Mendelian inheritance. The Gregor John Mendel, he gave certain laws, he proposed certain laws. This is how the diseases are going to be transmitted. This is how the genes are going to be transmitted from one parent to offspring. Okay, so Mendel's laws of inheritance. So those disorders which are following the Mendel's laws of inheritance. Okay, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. See, the point which I want to put into your mind is all this autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and even Y-linked diseases. They all follow Mendelian laws of inheritance. And these are the Mendelian disorders where there are point mutations. In all these conditions, there are point mutations. Okay, in almost most of them, there are point mutations. Okay, there is one base pair replacement. Now, there are certain diseases which do not follow Mendel's laws of inheritance. These are special. They do not follow Mendel's laws of inheritance. We will discuss. And there are chromosomal disorders, not just the genes, the entire chromosome is deleted, or half of the chromosome, Q arm is deleted, or there is mixing between 2 chromosomes, chromosomal disorders. For example, Down syndrome. What exactly is Down syndrome? Sir, Down syndrome is trisomy 21. Okay, trisomy 21, which means it's a chromosomal disorder. One extra chromosome came. Turner's. What exactly is Turner's? 45X0. X0, 0 stands for one chromosome is lost. X chromosome is lost. One X chromosome is lost. So they are the chromosomal disorders. Turner's, Edwards, Patau, Klinefelter's. Okay, these are all the chromosomal disorders. Now, multifactorial disorders. What exactly are the multifactorial disorders? Do you guys have any idea what are the multifactorial disorders? What are multifactorial disorders? Multifactorial disorders means there are diseases which just do not depend on the genes. They are not just dependent on the genes, they are dependent on the multiple factors. Okay, they are dependent on the multiple factors. For example, hypertension. Okay, hypertension, sir. See, hypertension, it is not only dependent on genes, but also on the other factors. Like, hypertension is the one thing which is not only dependent on the genes. Yes, of course, it is a familial, yes, possibility is there, but the patient's lifestyle, how much salt intake, the patient's food habits, the patient's lifestyle, smoking, alcoholism, or the patient's stress, the kind of environment he is working in, they are all going to determine whether the patient is going to develop hypertension or not. Okay, so a disease or a disorder, hypertension, it is dependent not only on the genes but also on other factors like lifestyle modifications, lifestyle, the food habits, the kind of environment he is working in. Okay, so these are the multifactorial diseases. Now, in your exam, the question that will come is, which of the following diseases follows? Which of the following diseases follows multifactorial inheritance? Okay, which of the following diseases follows? Which of the following diseases follows multifactorial inheritance? Now, can you tell me? This is the question. Multifactorial inheritance is followed by diabetes mellitus, hypertension. Okay, diabetes mellitus, hypertension, cleft lip, and cleft palate. This is the question that was asked in the PG exams. So which of the following diseases have multifactorial inheritance? Diabetes mellitus. See, diabetes mellitus is not only dependent on the genes. If the patient is sedentary, he is not working, he is taking excessive amount of sugars, more chances that he will get into the diabetes. Okay. Now, even though the patient is having certain genes for the diabetes, which are like mainly a risk factor for diabetes. See, imagine I'm the person who is having the genes which can take me to the diabetes, but with the kind of lifestyle I'm having, okay, I can, okay, with the kind of lifestyle that I'm having, I can go into diabetes very quickly. For example, if I'm a sedentary person, I'm taking excessive amount of sugars, I'm not doing exercise, I'm taking all kind of street foods, I will get into diabetes more quickly. Okay, so that's what I mean by. Now, anyway, after this, let's discuss about the autosomal dominant disorders. But wait, just tell me, how many types of genetic disorders we are going to discuss in this chapter? We are going to discuss about Mendelian disorders, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. Here, in your PG exams, this was a question asked, Y-linked inheritance pattern. Okay, Y-linked inheritance pattern is also called as Hollandric inheritance. Okay, so Hollandric inheritance or Y-linked inheritance. That's the question which I want you to know. Next, autosomal dominant. See, these autosomal dominant disorders, what exactly are they? See, let me give you examples later. First, let me tell you basic things. The most common mode of inheritance out of X-linked dominant, X-linked recessive, autosomal dominant, autosomal recessive, and Y-linked, out of all this, what is the most common inheritance pattern? The most of the diseases follow autosomal dominant pattern. Now, in this autosomal dominance, first you need to understand the concept of penetrance. So what will be the penetrance? The penetrance here is incomplete. There will be incomplete penetrance. Okay, see, these disorders, they will have incomplete penetrance. And even in these conditions, the expressivity in autosomal dominant condition is going to be variable or wide expressivity is seen. Now, most of the students, they don't have any idea. They don't have a proper idea about what exactly is the penetrance and what exactly is the expressivity. Okay, let me give you examples. First, see the definition. Later, I will give you the example. With that example, you will have the clear-cut idea. You will get a clarity. Now, look, what is a penetrance, sir? Penetrance means, okay, you know, in autosomal dominant disorders, the penetrance is incomplete penetrance. The expressivity is also incomplete. Wide variable expressivity is seen. Which means, first, let's discuss about the penetrance. Sir, penetrance is how often does someone with the genotype actually shows the corresponding phenotype. Okay, how often those people with a particular type of genotype will show the corresponding phenotype. Looking little complicated, right? Okay, looking little complicated, right? For example, look at this example. Okay, for example, look at this. This is a very funny example which I have find it out in the internet. Now imagine you are doing some experiment. Okay, you are doing some research. Okay, on 100 people. Okay, on 100 people. Now in this 100 people, you came to know that there are this 18. Okay, there are 3 rows, 6 in each. These 18 people are having a mutation. They are having a mutation, particular homozygous mutation. For example, capital R. They are having mutation. Okay, for this gene, particular gene, they are having a mutation. Okay, now see, these 18 people are having mutations. Sir. All these 18 people are having the same mutation. Okay, now, okay, let me put it this way. YRR, easy for easy understanding, you can say they are having this mutation. Okay, now, see, dekho. In these 18 persons, what exactly is happening? Look at their ears. Now, it's a mutation for the ears. Now, some of these people are having a very big rabbit kind of ears, big ears, or rat kind of ears. Okay. Now, tell me, in all these peoples, they are having the same mutation, but look, everyone is getting affected. No. See, these people, they are having normal ears. There are these people who are not having normal years, sir. So in all these people, same mutation is there, same homozygous mutation is there, but all of them are not having the disease. All of them are not having the same phenotype. All of them are not having the same phenotype. So this is called as a penetrance. Now in this condition, if everyone is affected, I will say penetrance is complete penetrance, I would have said. But in this condition, the penetrance is not complete. The penetrance is incomplete because even after having a defective gene, still the patients are normal. Some of the patients are normal. So now look at this definition. How often does someone with a genotype, okay, how often does someone with the same genotype, these are all the people with the same genotype, shows corresponding phenotype? Okay, now look here. Example, sir, out of 18 people, 11 people, see, out of 18 people, 11 people are showing the phenotype. Okay, they are all having this big ears. So how much is the 11 out of 18 are affected? Okay, even though after having the same genes, 11 out of 18 are affected. So what is the penetrance? Penetrance is 61%. Incomplete penetrance. So such a type of penetrance is shown by autosomal dominant disorders. If the patient is having autosomal dominant disorders, then the penetrance, even though the patient is having some defective genes, still they do not express the disease. This is called as penetrance. Now after this, look at the expressivity. Look at the expressivity. So what exactly is expressivity, sir? Expressivity means, okay, this is our expressive. Okay, expressivity. Expressivity means variation, variation in severity. Variations in the severity are expression of a phenotype. These are the two definitions you can understand. Expressivity means variations in the severity. Now, in this example, sir, yes, you know, the 16 individuals having the same type of genetic mutation. Even though after having the same type of genetic mutation, only 11 people are showing the disease. So penetrance is 61%. Penetrance is incomplete. But even look at the affected individuals. Sir, these affected individuals, see, okay, these affected individuals, now in this condition, look, in this example I am giving, these are the affected individuals. Now, are they having the same size ears in this example? Yes, sir, they are all having the same size ears. So, what is the expressivity? Sir, here the expressivity is wide range of expressivity is there? No, there is a narrow range of expressivity. All are having the same type of expression. All are having the same type of phenotype. Sir, you can say here gene mutation is there. All the patients who are affected are having same type of phenotype. But look here in this example. Sir, in this example, look, that this patient is affected having a big size ears. Now, these patients are affected who are having small size ears. Okay, so even after having same type of genetic mutation, even after having same type of genes, the expression, the phenotype is different. Different types of phenotypes are getting expressed. Okay, so this is called as variable expressivity or variable expression. Okay, variable expression. Now, let me give you one example. For example, imagine, simple example I will give you. Diabetes mellitus. Okay, diabetes mellitus. Of course, it's a multifactorial inherited disease. Now, diabetes, if I am the patient who is having a diabetes and there is some other patient who is having a diabetes, do you think, see, we are going to have the same kind of clinical features exactly in both the patients? No. I might be having vasculopathy, he might be having nephropathy, he might be having some other, problems. Even tuberous sclerosis also. Okay, there is one more disorder called as a tuberous sclerosis. For example, you can take any autosomal dominant disorder. Now I am having autosomal dominant disorder. The other person is also having the same type of autosomal dominant disorder, but still the symptoms in me and the symptoms in them, they can be different. They can be different. We are having the same diseases. We are having the same genetic mutations, but the clinical features can be different. That is called as the expressivity. Okay. So now I hope you have understood. So the penetrance and expressivity, they are the both different things. Penetrance is how often they will show the disease. Expressivity is how differently they are having the symptoms. Okay, so this is what I want you to know. Now, after this, what else you need to know? The most important topic now is autosomal dominant disorder examples. Autosomal dominant disorders. What are the examples of autosomal dominant disorders? The one thing you already know. The autosomal dominant disorders, they show incomplete parent trends and variable expressivity. Okay, well and good. Now, what are the examples? Any idea, guys? Autosomal dominant disorders, they include, let me write here. There are actually 15. I used to remember something like this. Okay, anything which starts with a hereditary, anything which starts with a hereditary, they are almost comes under autosomal dominant. Our family is the most important thing for us, dominant thing for us, our family. So autosomal dominant, anything starts with a family. Okay, familial, autosomal dominant, sir. Okay, let me write first here. Hereditary. Okay, hereditary. Spherocytosis. Spherocytosis. Hereditary spherocytosis. Next, hereditary cholesterolemia. Okay. Next, hereditary non-polyposis colon cancer, HNPCC, which is also called as Lynch syndrome. Okay, hereditary spherocytosis, hereditary hypercholesterolemia, hereditary non-polyposis colon cancer, colorectal cancer, which is also called as a Lynch syndrome. Okay, important for your exams. Now here, the recent question which was asked in the exam is, in hereditary spherocytosis, which protein defect, can you tell me what is the protein defect in hereditary spherocytosis, where the RBCs will become spherical? RBCs will become spherical, sir. Hereditary spherocytosis. Do you guys have any idea? Hereditary spherocytosis, where RBCs are getting round in shape. The defect is in ankyrin. Okay, MCQ which you should know. Next, so hereditary spherocytosis, Lynch syndrome, hereditary non-polyposis colon cancer. Next, hereditary telangiectasias, and so these are the hereditary things which are completed. Next, I have said you familial. Our family is a dominant thing. Okay, so familial adenomatosis polyposis. Familial adenomatous polyposis. FAP. Next, familial hypercholesterolemias. Okay, familial hypercholesterolemia. Next, I used to remember something like this. Sir, hunting. Okay, hunting is a dominant sport, right? Hunting is a dominant sport. So something like Huntington's disease. Okay. Huntington disease. Next, a very famous MCQ about the kidneys. Okay, about our kidneys. There is one problem which is going to occur to our kidneys, which is autosomal dominant inheritance pattern. Do you know what is that? It starts with the letter AD only. That is ADPKD. Okay, autosomal dominant polycystic kidney disease, which is adult in onset. Okay, adult onset polycystic kidney disease. Adult onset polycystic kidney disease, also called as ADPKD. It is also autosomal dominant disorder. Next, kidney completed. Okay, kidney completed. Ninth, heart related problem. Heart, dominant thing. What? Heart is getting dominant, bigger and bigger. What is that? Hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy, sir. Hypertrophic cardiomyopathy. So it is also following autosomal dominant pattern of inheritance. Now apart from that, okay, now hunting completed. See, simple. Anything which starts with the letter hereditary, hereditary spherocytosis, hereditary hypercholesterolemias, hereditary non-polyposis colorectal cancer, hereditary telangiectasias, or anything starts with the familial also. Okay, so those are all autosomal dominant. Now after that, Huntington disease, hunting, dominant sport. Now autosomal kidney problem, autosomal dominant kidney disease, hypertrophic, big heart. Okay, dominant thing, hypertrophic cardiomyopathy. Now bone problems, two bone problems I want you to know, which are called as osteogenesis imperfecta. Okay, osteogenesis imperfecta, as well as achondroplasia, cartilage problem. Achondroplasia is actually like the cartilage thing, right? So achondroplasia Okay, achondroplasia. Yes, if you have any doubts, you can put. Okay, achondroplasia, osteogenesis imperfecta. Okay, osteogenesis imperfecta. These are the autosomal dominant disorders. They are also following the autosomal dominant inheritance pattern. So apart from that, there are some miscellaneous disorders which you need to by heart. Okay, which you need to by heart for sure. What are they? Leaf-Fermani syndrome, Noonan syndrome, so Leaf-Fermani syndrome, Noonan syndrome, and muscular dystonia, muscle. Okay, muscle dystonia, and last and final thing, that is pseudo hypoparathyroidism. In pathology, in the parathyroid topic, we'll discuss about hypoparathyroidism, primary hypoparathyroidism, secondary hypoparathyroidism, pseudo hypoparathyroidism. Okay, pseudo hypoparathyroidism. We'll discuss about them. But right now the point is pseudo hypoparathyroidism. So they are all examples. So 15 completed, sir. Okay. So there are 15 autosomal dominant disorders, which I want you to know is, just don't look at there and just try to answer. What are they? All hereditary things. What are all the hereditary things? Hereditary non-polyposis colorectal cancer, HPC, hereditary non-polyposis colorectal cancer or colon cancer, which is also called as the Lynch syndrome, One thing, hereditary hypercholesterolemias. Okay, next, hereditary spherocytosis, where there is defect in the spectrin, and hereditary, last one, telangiectasias. Telangiectasias means blood vessel problem. Okay, dilation of the blood vessels. Next, familial adenomatous polyposis. Okay, familial adenomatous polyposis. And next, Huntington disease, autosomal dominant polycystic kidney disease, hypertrophic cardiomyopathies, achondroplasia, osteogenesis imperfecta, Leaf-Fermani syndrome, Noonan syndrome, muscular dystonia, and last and final thing, it is the everything hypertrophic cardiomyopathy. All these are the, sorry, last one is not, already I have discussed hypertrophic cardiomyopathy. The last one, pseudo hypoparathyroidism. So these are the autosomal dominant disorders. Now one point which I want you to know is, see, if you are having any of these disorders, autosomal dominant disorders, from when you will get these diseases? From birth or from adolescence? See, these diseases will start to manifest in the body from adolescence or from the adulthood. Okay. So diseases are going to be seen. Disease usually manifest from adolescence. Okay, from the adolescence. These are not childhood onset. Autosomal recessive disorders are childhood onset, but these are not autosomal recessive. These are all autosomal dominant. 15 disorders I gave you. One thing will come in your exam for sure. If you know this 15, more than enough. You need to by heart it. There are many mnemonics which are available in the Google. You can go through them. But see, again you have to remember that mnemonic also. Remembering this 15 names itself, it is difficult. So again remembering a mnemonic is a kind of difficult thing. So just by heart them, sir. Now after autosomal dominant disorders, let me give you examples for autosomal recessive disorders. Autosomal, right? Autosomal recessive disorders. Now there are 15 examples for autosomal dominant. Now you have to know 15 examples for autosomal recessive also. But before telling you, let me tell you something. These are childhood onset. In the next class, I will discuss about the pedigree pattern, how the pedigree questions that will come in your exam. Okay, what is the how to identify the pedigree for autosomal dominant disorders, autosomal recessive, X-linked dominant, X-linked recessive, mitochondrial diseases, how to identify the pedigree. That we will discuss in the next class. In this class, just the examples. Okay, examples part. They are going to be childhood onset disorders. They are going to be there from childhood onwards. Second thing, penetrance. So what about the penetrance? The penetrance is complete. Okay, there are 100 individuals who are having these defective genes. Every person is going to manifest the disease. Every person is going to show the disease. Complete penetrance. It's not like, see, autosomal dominant disorders follow incomplete penetrance, which means even though some individuals are having the defective genes, still they will not manifest the disease. They will not show the disease. But autosomal recessive disorders are not like that. They will have complete penetrance. And expressivity is also very narrow. Okay, narrow expressivity. Okay, which means if 100 individuals are having the disease, means all 100 individuals will show the same type of symptoms. There is no variability. There is no variability, right? All patients will show same symptoms. Okay, so expressivity. Now, let's do the examples. See, let me write the examples. Phenylketonuria. I used to remember something like this. Anything which starts with the ends with the letter urea. Phenylketonuria, alkaptonuria, homocystinuria, anything which ends with the word urea, it's autosomal recessive. Okay, so phenyl, ketone, urea. Phenylketonuria, alkaptonuria, homocystinuria. Okay, these are all examples of autosomal recessive disorders. See, out of 15, 3 are completed. Okay, out of 15, 3 are completed. Now, what are the others, sir? Now, let me write one anemia. Which type of anemia? Sickle cell anemia. Sickle cell anemia. Okay, so sickle cell anemia, missense mutation, glutamic acid is replaced with valine. So that example, sir, sickle cell anemia. Next, there is one disorder where a female child will look like a male. A female child is going to look like a male, sir. That is female pseudo hermaphroditism. What is that condition? Congenital adrenal hyperplasia. So congenital adrenal hyperplasia, deficiency of enzyme 21 hydroxylase and 11 beta hydroxylase. So that syndrome, sir. Okay, congenital adrenal hyperplasia. See, important points about this autosomal dominant disorders. For example, important points what you need to know regarding congenital adrenal hyperplasia will be discussed in the next video. Okay, even some important autosomal dominant. Okay, let me write one important thing I forgot here. In autosomal dominant, add one more thing. 16th, Marfan syndrome. Marfan syndrome, MCQ. This is very important, sir. Marfan syndrome. Next, 17th, neurofibromatosis. Neurofibromatosis. Okay, Marfan syndrome, neurofibromatosis also follows the autosomal dominant inheritance pattern. Okay, next, let's come to the autosomal recessive. What we have completed: phenylketonuria, alkaptonuria, homocystinuria, sickle cell anemia, congenital adrenal hyperplasia. Next, Friedreich ataxia. Okay, Friedreich ataxia. Next, any other disorder, where there is excessive deposition of copper in the body. There is this one disorder where there is excessive deposition of copper in the body, which is called as Wilson disease. Wilson's disease. And excessive deposition of iron in the body, that's the hemochromatosis. Okay, hemochromatosis, sir. So Wilson's disease, important points about the Wilson's disease, that's celluroplasmin deficiency is there, the Kaiser-Fleischer rings will be seen in the eyes, and hemochromatosis, excessive iron deposition in the body, the iron is stained with the pearls Prussian blue. Look, Marfan syndrome, what are the important points which you need to know? Neurofibromatosis, what are the important points which you need to know? Okay, and let me add one more autosomal dominant disorder, that is tuberosclerosis. Okay, tuberosclerosis. This is also autosomal dominant disorder. So I will discuss about this autosomal dominant disorders, important autosomal dominant disorders which you need to know for your exams. Oh, this is the important MCQ about the Marfan syndrome. This is the important MCQ about the phenylketonuria, something like that. Okay, so phenylketonuria, alkaptonuria, homocystinuria, important points are there. Autosomal recessive disorders, I will discuss in the next class, sir. Okay, because I'm little irritation is there in the nose, so I will complete it quickly. So hemochromatosis, Wilson's disease completed. Now after that, see all the inborn errors of metabolism, like the lysosomal storage diseases, lysosomal storage. You know, right? Niemann-Pick disease, Gaucher's disease. Okay, those diseases, lysosomal storage disorders, and even glycogen storage disorders. Lysosomal storage disorders and glycogen storage disorders are all these disorders, lysosomal storage disorders, glycogen storage disorders, they are all examples of autosomal recessive disorders. Now next, one more thing, cystic fibrosis. Cystic fibrosis, you know it, right? CFTR gene mutation, thick mucuses are going to be produced in the body, leading to the obstruction of the ducts. So cystic fibrosis leading to pancreatitis, acute pancreatitis, and even bronchiectasis in the lungs. So cystic fibrosis. Next, ataxia, telangiectasia. Okay, next, Fanconi's anemia. See, these are all examples. Okay, these are all examples of autosomal recessive disorders. Any other examples? Yes, xeroderma, pigmentosa. Next, a syndrome called as a Bloom syndrome. So these are examples which you need to know for your exam. If you know this much, that's more than enough, sir. Okay, so like, see, just recap. What are the examples of autosomal recessive disorders? The examples of the autosomal recessive disorders are phenylketonuria, alkaptonuria, homocystinuria, sickle cell anemia, congenital adrenal hyperplasia. Now, one important point is albinism, right? Albinism. Okay, so albinism, one more example which is following the autosomal recessive inheritance pattern. Next, Friedreich's ataxia, Wilson's disease, hemochromatosis, all lysosomal storage disorders. Exceptions are there. Exceptions are there. Hunters is an exception. Okay, Hunters is an exception. We'll discuss later. Lysosomal storage disorders, glycogen storage disorders, cystic fibrosis, ataxia telangiectasias, Fanconi's anemia, xeroderma pigmentosa, Bloom syndrome. These are all examples of autosomal recessive disorders. Now, one more thing, alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin deficiency leading to emphysema. Okay, leading to emphysema. So this is also an example of autosomal recessive disorders. Okay, welcome back, guys. Now we are in the chapter of genetics. Already in the last class, we have discussed about the basics in the genetics. We have seen how many types of, how many number of genes are there, where exactly the genes are there. We have discussed about autosomal dominant, autosomal recessive disorders. We have discussed about the topics of penetrance, expressivity. We have discussed about the locus. See, these things we have discussed in the last class itself. Okay, and I have also discussed about the examples for the autosomal dominant and autosomal recessive disorders. Now, in the last class itself, I told you that I will be discussing some important points about the autosomal dominant disorders as well as autosomal recessive disorders. Okay. See, in the last class I got a little cold, so the class was like, okay, so kind of thing. So in this class, let's continue with the same autosomal dominant and autosomal recessive disorders. After that, we also have to complete the X-linked dominant and X-linked recessive disorders along with the Y-linked inheritance. Okay. So having said that, now in this class, let's begin with the autosomal dominant disorders. Which autosomal dominant disorders we have discussed in yesterday's class? We have discussed almost 15 disorders. I gave the examples. Okay, like anything which starts with the hereditary or anything which starts with the familial. Like we have discussed 15 disorders. Now out of those, the first disorder which I am going to discuss now is called as a Marfan syndrome. Okay, Marfan syndrome. Okay guys, welcome back. So now we are discussing about the Marfan syndrome. In yesterday's class, we have discussed Marfan syndrome, the tuberous sclerosis. Okay, these are all disorders, osteogenesis imperfecta. Okay, all these disorders are autosomal dominant disorders. Now, let's see some important points about the Marfan syndrome. Why? Because this Marfan syndrome is a very important topic for your exams. The Marfan syndrome, my question is, what is the gene defect? What is the gene that is defective in Marfan syndrome? So what is the gene defect? A question? Any idea? So the gene defect in Marfan syndrome is fibrillin gene. The fibrillin gene is defective. So when the fibrillin 1 gene is defective, what is the protein that's going to be affected? What is the protein that is affected in the Marfan syndrome? I used to remember something like Marfan, F for, like, Marfan, that F for fibrillin gene, and that protein which is defective is fibrillin protein. Okay, fibrillin protein is the defect. Okay, so the fibrillin protein is defective. So the gene affected is the fibrillin 1 gene, and the protein that is going to be defective, the protein that is defective here is the fibrillin. Okay. Now one important point which I want you to know here is, for your exam purpose, fibrillin is very much important. Fibrillin is very much important for the assembly of the elastin fibers. For the elastin to assemble, you need fibrillin, sir. Okay. So fibrillin is needed for the assembly of elastin fibers. So whenever this fibrillin is defective, automatically the elastin will also be affected. So elastin will be negatively affected. So directly affected is fibrillin. The fibrillin is directly affected, and also in this condition, elastin, okay, elastin protein is indirectly affected. On your exams, they can tell you which protein is negatively affected in this condition of Marfan syndrome. That is elastin. So directly affected is the fibrillin and negatively affected is the elastin. Now in this condition, what else you should know for your exams is, sir, in Marfan syndrome, okay, this fibrillin actually controls the levels of transforming growth factor beta. Okay, the transforming growth factor levels are controlled by the fibrillin. Now in this condition, the fibrillin is not there. Okay, fibrillin gene is defective and the fibrillin is not there. What happens to the transforming growth factor beta levels? The transforming growth factor beta levels, okay, let me write TGF beta. Okay, transforming growth factor beta levels are going to be elevated. Okay, very high. So in Marfan syndrome, they can ask in your exam, which levels are elevated? It's the transforming growth factor beta levels are elevated. Okay, now this elevation, okay, elevation in this transforming growth factor beta is the one, this is the one which causes the symptoms. I will discuss about the clinical features later. So now as a treatment part here, so in this condition of Marfan syndrome, the transforming growth factor beta levels are elevated. Okay, now what we have to do as a doctor, we have to decrease the transforming growth factor beta levels. So which drug, okay, that's the question, which drug can actually decrease the transforming growth factor beta levels? Come on guys, what is that one drug? What is the treatment? The treatment is, any idea? It's the losartan, sir. Inhibitors, TGF beta inhibitors, losartan. Okay, so losartan is used in the treatment of Marfan syndrome. Okay, done. Well, so now let's discuss about the clinical features. Clinical features of Marfan syndrome. Now, guys, can you tell me what are the clinical features of Marfan syndrome? There is a mnemonic, there is a very famous mnemonic, the Marfans, the mnemonic itself is a Marfans. Okay, so first let's write M. Okay, so what exactly M stands for? In Marfan syndrome, what exactly is the M stands for? M A R F A N S, Marfans. Here the M stands for mitral valve prolapse. Okay, mitral valve prolapse. Here let me just try to integrate. See, if I am the person, right now if I am the person who is having a mitral valve prolapse because of the Marfan syndrome, see, I'm the one who is having mitral valve prolapse. Now when you auscultate my heart sounds, now what is that thing? What is that thing which you can hear? What is that keyword? If I am the one who is having mitral valve prolapse, what is that murmur kind of thing that you can hear? Any idea? In mitral valve prolapse, any idea? Mid systolic clicks. Okay, So in the condition of mitral valve prolapse, the patient is going to have, yes, okay, which one? It's a mid-systolic click. Okay, mid-systolic click. Whenever you see the word mid-systolic click, think about the mitral valve prolapse. Now what is this A? A stands for aortic dissection. Okay, so aortic dissection means, see, it's a Marfan syndrome is a connective tissue disorder. First thing you should know, so this Marfan syndrome, it's a connective tissue disorder. See, in the connective tissue, which the fibers are going to be affected, you know it. Okay, which fibers are going to be affected? It's the fibrillin. Okay, fibrillin fibers and elastin fibers are going to be affected. Now, because of this, the connective tissue is weak. Even in the aorta, the connective tissue is weak. The aortic valve is now not having proper strength. Now, in this condition, what happens is the jet of blood can actually tear the aortic wall into 2 slices. Okay, so what happens in this Marfan syndrome is aortic dissection. The blood is now passing through the wall of the aorta. Okay, now imagine this is the aortic wall. Now the aortic wall is going to splits because of the weak connective tissue. So aortic dissection, the thing which can ask in your exams is why aortic dissection? Why? Because of cystic medial necrosis. So cystic medial necrosis is seen in Marfan syndrome. This can lead to the media of the blood vessels. Okay, the media of the blood vessels is undergoing necrosis. Okay, cystic medial degeneration is seen in Marfan syndrome that can lead to aortic dissection. Okay, now what is R stands for? Any idea? Retinal detachment. Okay, retinal detachment. And F stands for, see, I used to remember like F, Marfan syndrome, F for fibrillin 1 gene defect. Okay, fibrillin 1 gene defect. And A, what is the next A, sir? A stands for, if you look at these persons, their fingers are too much elongated. Okay, their fingers are too much elongated. Now, if you look at their fingers, I will show you the image. Their fingers are looking like a spider legs. Their fingers will look something like a spider legs. So, this is something called as arachnodactyly. Okay. Arachnodactyly. I will show you the image. And also, in this person, if you look into their mouth, the palate is going to be in arch shape. Okay. So, arched palate. Okay, the palate is going to be like an arch. I will show you the image. Okay, arched palate. And what's that N stands for? N stands for, so these patients are going to have nearsightedness. Okay, and last S stands for, any idea what is this S stands for? Love from Pakistan, love from India. Okay, what is that S stands for? Let me write here, subluxation. Subluxation of lens. Okay, subluxation of the lens. So, in these patients who are having the Marfan syndrome, the lens is going to be dislocated. The lens is a connective tissue defect, right? So, the lens is not going to stay in the proper place. The lens will be dislocated. That is called as subluxation or ectopia lentis. Okay, ectopia lentis. Now, where this lens is going to be dislocated and which side? That's also an MCQ in your exam. Which side the lens is going to be dislocated? Is it going to be dislocated superiorly, nasally, medially, or is it going to be dislocated inferiorly? Which side? See, the lens dislocation is, that's MCQ, superior temporal dislocation. So, there will be superior temporal dislocation of the lens. So, that's what is seen in the Marfan syndrome. For your exam, this is very important. Okay, superior from the aphthal. Okay, from the aphthal, superior temporal dislocation of the lens is seen in Marfan syndrome. But if they ask you inferonasal dislocation, okay, inferonasal dislocation. of lens. So now can anyone answer in which condition there is inferonasal dislocation? So the lens is again dislocated, but it is towards inferior side, inferior and nasal, inferonasal dislocation. It is seen in, any idea? Homocystinuria. Okay, homocystinuria, sir. Okay, so that's for the S, subluxation of the lens. The ectopia lent is superior temporal dislocation of the lens. And one more S, sir. One more S here I want to discuss is these patients will have scoliosis. Okay, these patients are going to have scoliosis. Okay, these patients are going to have scoliosis. And not only that, the last one which I want you to know is they have pigeon shaped chest. Okay, last P. Okay, Marfan P. I used to remember Marfan P. This pigeon shaped chest. Okay, you know, right? The patient is going to have a pigeon shaped chest. Then we are going to call this condition as pectus carinatum. Okay, pectus carinatum, the pigeon shaped chest. Not only that, pigeon shaped chest, even they might be having excavation also. Okay, pectus excavation, okay, pectus carinatum and pectus excavatum. Okay, so both the things are possible. Yeah, just remember, okay, for your exam purpose, okay, these persons can have pigeon shaped chest, which is called as pectus carinatum. Okay, pectus carinatum. I will show you the image. So these are some important clinical features which I want you to know. Now look here. So one more clinical feature I just want to add. These patients are going to be very tall. Okay, these patients are called as, these patients are going to look very tall, sir, especially their limbs. Their limbs are going to be very much, the length of the limbs is going to be very much in nature. So this is something called as Marfanoid habitus. Okay, this is something called as Marfanoid habitus. So these patients are going to be very thin and slender. These patients are going to be very thin and slender with the long extremities. Okay, with the long extremities, arm length is going to be very much long. So this is something called as a Marfanoid habitus. Now after this, you can very clearly see here. See, this patient is thin and slender. Now if you look at his arms, they are very long. They are very much lengthier. Okay, and even this female, you can very clearly see that she is having a scoliosis. Okay, scoliosis. She is having the scoliosis. And here, what else? I have told you these patients will be having some chest abnormality, that is either they can have a pigeon shaped chest, that is called as a pectus carinatum, or even they can have pectus excavatum. Okay, pectus excavatum, this is P, pectus excavatum, or even they can have pectus carinatum. Okay, both are possible, Okay. Now what else, sir? What else? Look at their the fingers. The fingers are going to be very lengthier. Okay, the fingers are going to be very much lengthier. Now what you can see here, so this is high arched palate. There is high arched palate, and you can very clearly see here there is superior temporal dislocation of the lens. Okay, superior temporal dislocation of the lens. Okay, that's one thing. And what else, sir? What else? See, these patients, I have told you, these patients aorta problems. What is that aorta problem? See, first initially there will be aortic root dilatation. Okay, aortic root dilatation can be seen initially. See, there is aortic aneurysm, aortic root dilatation, followed by later on they will have aortic dissection. So both things are possible. Okay, let me write here. So aortic dissection, and not only that, aortic root dilation. Okay, aortic root dilation and aortic aneurysm. Okay, aortic aneurysm, sir. And later on they can also develop the aortic dissection. Okay, now here you can very clearly see. So these patients, look at their fingers, how their fingers are. So these fingers are very long fingers, and they are called as what? They are called as arachnodactyly. Okay, arachnodactyly. Even the legs you can see the same thing. And here you can very clearly see that's a pigeon shaped chest, and the patient is going to have scoliosis. Okay, scoliosis. So these are some important points which I want you to know regarding the Marfan syndrome. Now in this Marfan syndrome patients, you can appreciate two signs. What are those two signs? Look here. Now first this one. Okay, normally when, see, this is my wrist. Okay, this is my wrist. When I do like this, see, hardly I can bring my fingers close together. Okay, hardly. Okay, just that touching. Okay, but in these patients who are suffering with Marfan syndrome, they go, see, their fingers are very much lengthier. They are tall, like the fingers are very long fingers. Okay, long fingers. So they can overlap. The fingers are going to be overlapped even after the, when you put it at the wrist joint, the fingers will overlap. The thumb and the fingers will overlap, sir. Okay, that's the one point which I want you to know. Okay, the fingers will overlap, especially the thumb and the little finger. Now see, thumb and little finger, they will hardly meet. But in these patients, because the fingers are long, okay, they will overlap. The thumb and even little finger will overlap, sir. So this sign is called as a Walker sign. So now tell me, Walker sign is seen in which condition? Walker sign is seen in Marfan syndrome, sir. Marfan syndrome. And not only that, see, when you try to close your wrist, when you try to close the palm like this, now the thumb will never be exposed out. The thumb will never be exposed out. See, in a normal healthy person, now when you close your thumb, you close your fingers like this, the thumb will never come out. You cannot see it. Okay, hardly in some persons you can see a little bit amount of thumb. But here, look in this condition, as their fingers are tall, the large portion of thumb is being exposed to outside. So this sign is called as Steenberg sign. Okay, Steenberg sign. So Steenberg sign is again seen in which condition? It is seen in Marfan's syndrome. Okay, so Steenberg sign and Walker sign. Now just tell me, what is this? What you are looking at here? Here the person is having dislocation of the lens. Dislocation of the lens seen in Marfan syndrome, sir. Okay. Now after this, let me show you a table. So here, from this table, you can actually, what you should know is, so how to put the diagnosis. Okay, how to put the diagnosis of the Marfan syndrome. Okay, how to put the diagnosis of the Marfan syndrome is, see, there are major criteria which you should know. See, I have gave you so many clinical features, right? There are so many clinical features I gave you. Now, what are the major criteria? Okay, here. See, the major criteria are ectopia lentis, superior temporal dislocation of the lens. That's one of the major criteria. Aortic dilatation or aortic dissection, I have discussed. That's one of the major criteria. And the family history, positive family history. Why? Because it's an autosomal dominant disorder. As it's an autosomal dominant disorder, definitely the bloodline, definitely the father or the mother. Okay, in the family history, definitely there will be one person who is suffering with Marfan syndrome. So, family history, that is coming positive. Ectopia lentis, along with aortic root dilatation or the aortic dissection, they are the three major criteria. Okay, remaining all of them are the minor criteria, sir. Okay, remaining all of them, they are the minor criteria which are considered as the systemic score. Okay, we'll give for everything, we will give 1 point. Okay, no need to go into that much detail. Only for your exam, just remember what are the major criteria: ectopia lentis, aortic dissection, and family history. Now you know the other important points. What is the mode of inheritance of this Marfan syndrome? Mode of inheritance is autosomal dominant. Okay, autosomal dominant, fibrillin 1 gene mutation. FBN means fibrillin 1 gene mutation. Okay. Next, what you should know is, sir, what is the most common cardiac defect? Sir, what is the most common cardiac defect in this condition? The most common cardiac defect is mitral valve prolapse. Already you have, you know it. Already I have explained mitral valve prolapse. Okay, mitral valve prolapse, sir. Next, what else you should know here is, what is the criteria? Okay, criteria for the diagnosis. The criteria for the diagnosis for the PG aspirants, this is MCQ, Gens criteria. So, Gens criteria is the criteria used for diagnosis. So, according to the Gens criteria, see, you need to have two major criteria. Any two out of these three are one major plus fibrillin gene mutation. Okay, one major, okay, along with when you do the gene assays, the fibrillin gene is getting affected. So, at least that thing, or at least one major criteria out of these three, at least one major criteria or a systemic score greater than 7. So, what is this systemic score? Here it is there. Okay, no need to go in that much detail. Simple. For your exams, those who are going for the NEET PG exams, they should know only this thing. Gens criteria. Okay, this area. Gens criteria, it is used for the diagnosis of Marfan syndrome. Okay, modified Gens, revised Gens criteria. Okay. Next, what is the cause of death? What is the most common cause of death? The most common cause of death is cardiovascular complication. And what is that cardiovascular complication? It is the most common cause of death in Marfan syndrome is aortic, excellent fun mode, aortic dissection. Okay, aortic dissection. Done. So, if you know these many points about the Marfan syndrome, the first autosomal dominant disorder, that will be more than enough. Okay, that will be more than enough. Okay, definitely image based questions that might come in your exam. Now after this, let's discuss about this disorder. So look here and tell me, guys, can you tell me what is this image based question, what you are looking at? So these patients are having this small papule kind of things. What are these? Okay, what are these tumor kind of things? What exactly are they? So that is, yes, neurofibromas. Those are the neurofibromas. So the next disorder which we are going to discuss is the neurofibromatosis. Okay, neurofibromatosis. Now my question to you is, how many types of neurofibromatosis are there? How many types of neurofibromatosis? There are two types. Neurofibromatosis type 1 and neurofibromatosis type 2. Neurofibromatosis type 1 as well as neurofibromatosis type 2. Now, questions. Sir, in neurofibromatosis type 1, first of all, now wait. Sir, why the hell are we discussing about neurofibromatosis? Now you can ask me, sir, why we have discussed about the Marfan syndrome? Because Marfan is autosomal dominant disorder. Important autosomal dominant disorder. Important for what? Important for your exams. Now, why we are discussing about neurofibromatosis? Neurofibromatosis also autosomal dominant disorder. Okay, that's why we are discussing. Now, there are two types of neurofibromatosis type 1 and neurofibromatosis type 2. Yes, Monica, yes, chromosome number 17 as well as, excellent. Okay, so neurofibromatosis, one gene. Okay, the neurofibromatosis type 1 is because of defect in neurofibromatosis one gene. Okay, neurofibromatosis one gene is defective. The neurofibromatosis one gene is present on chromosome number 17 and neurofibromatosis type 2 gene is present on chromosome number 22. So, NF1 and NF2 genes are mutated. These genes are present on chromosome numbers 17 and 22. Now, okay sir, you know the gene defect. So, because of the defect in these genes, which proteins are going to be affected? Sir? In NF1, neurofibromatosis type 1, can you tell me? It is a neurofibromin. Okay, neurofibromin is a protein defective. And in neurofibromatosis type 2, it's a Mendelian. Okay, Mendelian is a protein that is affected. Now, sir, in neurofibromatosis, what are the clinical features? Okay, there are two types of neurofibromatosis. In both the conditions, the patient is going to have this kind of macules, this kind of spots on their skin. Now, what is this spot, sir? So, what is this spot that you are able to appreciate here, or that you are seeing here? What is this spot on the skin? This is something called as, that is cafe allele spots. Okay, so these cafe allele spots are seen, just tell me, in type 1 neurofibromatosis or type 2 fibromatosis? It is seen in both. Okay, both in neurofibromatosis type 1 and type 2, both in type 1 and type 2. Now after that, what you are able to see here? Okay, what you are able to look in the screen? So these are the eyes. Okay, these are the eyes. Now in this eyes, you can see these pigmented areas in the iris. Okay, there are these pigmented areas in this iris. So what are these? Can you tell me? These pigmented areas that you can see in the iris. So these are the leash nodules. Leash nodules. So these leash nodules are seen in only type 1 neurofibromatosis. MCQ. Leash nodules are seen in neurofibromatosis. Okay. Now more high yield question. Leash nodules are seen in which type of neurofibromatosis? Only seen in type 1 neurofibromatosis. Okay, only seen in type 1 neurofibromatosis, sir. Now, apart from that, sir, in type 2, okay, in type 2 neurofibromatosis, what are some important points, sir? In type 2, what you will see? Actually, let me write here. In type 2 neurofibromatosis, the patient is going to have some important tumors. What are they? Schwannomas, acoustic schwannomas. Okay, acoustic schwannomas are going to be seen. Now, in this acoustic schwannoma, acoustic audition, in this acoustic schwannoma, which cranial nerve is going to be affected? So, these acoustic schwannomas are going to occur for which cranial nerve? The 8th cranial nerve. That is vestibulocochlear nerve, 8th cranial nerve. And the patients are also going to have meningiomas, Okay, and the patients are also going to have anything else? Ependymomas. Okay, ependymomas. So these are the three types of tumors. Okay, if a patient is suffering with a neurofibromatosis type 2, he is going to have bilateral acoustic schwannomas, where the eighth cranial nerve is going to be affected. The patients are also going to have meningiomas and ependymomas. Why all these problems? Because of neurofibromatosis type 2 gene, which is present on the chromosome number 22, it is mutated. Because of that, the Mendelian protein is again gone. Mendelian protein is defective. When the Mendelian protein is affected, it is going to cause neurofibromatosis type 2. And one important point regarding meningiomas, never ever forget that the patients are going to have meningiomas, sir. Meningiomas, will see which bodies? Samoma bodies. Okay, meningiomas, samoma bodies are seen. Ependymomas, what you will see? In ependymomas, there will be drop metastasis. Okay, ependymoma, it's a cancer, right? It's a cancer. Ependymoma is a cancer. Now it will show drop metastasis. Okay, drop metastasis. Okay, so these tumor cells, they will drop into the cerebrospinal fluid. Okay, these tumor cells, they will drop into the cerebrospinal fluid and they will show the metastasis, the drop metastasis. Very important MCQ. Which tumors will show drop metastasis? Ependymomas. Question. Okay, now list nodules completed. Now you can very clearly see here. See, neurofibromatosis type 1. Okay, now what and all will be seen? We know, we have already discussed, sir, the patients are going to have skin involvement is a neurofibromas. This kind of a neurofibromas, these tumors are going to be seen. Okay, sir, and there will be cafe spots are going to be seen, and the patient is going to have this pigmented hamartomas, the pigmented hamartomas in the iris, which are called as the Lisch nodules. Okay, these Lisch nodules, they are called as pigmented hamartomas. Okay, they are the pigmented hamartomas in the iris. Now, apart from that, so is there any other thing which you need to keep in your mind? Okay, yes, they are there. See, the patients are going to have, yes, cafe spots are there, and the skin neurofibromas are there. And not only that, in neurofibromatosis, this is a dermatology question, there will be axillary frickling. Okay, there will be axillary frickling, freckles. Okay, freckles, small, axillary frickling will be seen. Okay, axillary frickling is going to be seen, sir. These are the neurofibromas and these are the cafe spots. Now, apart from that, here you can very clearly see the patient is going to have the Lisch nodules, which are the pigmented hamartomas in the iris. And one important point which I don't want to miss here is optic nerve gliomas. These patients are going to have the optic nerve involvement, optic nerve gliomas. Okay, let me write here. The patient is also going to have optic nerve gliomas. Okay, and in the future these patients are at risk of developing breast cancer also. But anyway, important points about the neurofibromatosis completed. Okay, never forget about these proteins and neurofibromin as well as Mendelian. Chromosome number 17, chromosome number 22. 17, 22. Okay, sir? Yeah. Now, after this, let's discuss about the third autosomal disorder, which is called as tuberosclerosis. Okay, tuberosclerosis. Come on, guys. What is the gene mutation? Do you know? Do you know what is the gene mutation seen in the tuberosclerosis? Any idea? Tuberosclerosis, what is the gene mutation? The gene mutation is TSC, tuberosclerosis gene. Okay, TSC gene mutation. Sir, there are two types of TSC genes, TSC1 and TSC2 gene. TSC1 and TSC2 gene. The TSC1 gene and TSC2 gene, they are coding for, okay, they are coding for which protein? They are coding for hamartin, and tuberin. Okay, tuberin, hamartin and tuberin. So MCQ, sir. So TSC1 gene is coding for a protein called as hamartin. TSC2 gene is going to code for a protein called as a tuberin. Now, because of the mutation of this TSC gene, the patient is going to have tuberosclerosis. Now, what are the important things which you need to know regarding the tuberosclerosis, sir? Very important for FMGE exam as well as the NEET PG exam. See, the patients are going to have something, the symptoms are, remember it like a ash leaf. Okay, ash leaf. ASH LEAF, that's a mnemonic, sir. Now, when you look at these patients are going to have ash leaf spots on their skin. Okay, ash leaf spots. Now, what are these ash leaf spots, if you ask me? Sir, look here, this white color. Okay, this white color spots which are seen. These are not hyperpigmented, these are hypopigmented. They will ask you. Ash leaf spots, these macules, ash leaf macules, they are hypopigmented lesions on the skin. Okay, sir. And not only that, this thick leathery patch like appearance on the lumbar region, this is called as a Sjogren's patch. Okay, ash leaf spots are going to be seen, Sjogren's patch, and whenever the patient is having this TSC1 gene or TSC2 gene mutation, the patients are going to get this tuberosclerosis, where the patients are going to have the cardiac involvement, that's the rhabdomyomas. Okay, the tumors of the heart, the rhabdomyomas, and in the lungs, okay, L stands for lung, lymphangioliomyomatosis. Okay, lymphangioliomyomatosis. And E for epilepsy, because of the cortical tubers. In the cortex also these tubers will come. The tuberosclerosis, the patient is going to have the tubers. Okay, small tubers. Tubers are nothing but the growths, like tumor like growths. Okay, and this is very important. Angiolipomas in the kidney. Angiolipomas, when we are discussing about the renal pathology, then we will discuss, sir, in the kidneys different types of tumors. First tumor that we will discuss in the kidney is angiolipomas. Okay, so angiolipomas are associated with which condition? Okay, angiolipomas are associated with the tuberosclerosis, and finally facial angiofibromas. Okay, see here, the patient is having an angiofibroma, that's a facial angiofibroma. Here you can very clearly see this thick leathery patch which is present on the lumbar region, that's called as a Sjogren's patch. You can see the cortical tubers can be seen, that can lead to epilepsy, and here also the patient is having these tubers. Okay, small tubers, these are the tuberosclerosis, small tubers will start to form. Okay, so now we have discussed the three important autosomal dominant disorders which are important for your exam. Okay, so tuberosclerosis, next neurofibromatosis, and Marfan syndrome. Okay, Marfan syndrome. With this, autosomal dominant disorders are done. Okay, autosomal dominant disorders. I have given 15 autosomal dominant disorders, but I have selected 3 autosomal dominant disorders which will come in your exam. Okay, now after this, let's discuss about the autosomal recessive disorders. Okay, autosomal recessive disorders. Guys, shall we continue? Autosomal recessive disorders. Now, say first thing. Okay, MBBS, first thing remember, whenever you see about any inborn errors of metabolism, right? Inborn errors. Okay, inborn errors of metabolism. What are the inborn errors of the metabolism? The metabolic reactions, sir. Like the metabolic reactions are not happening properly. Like they are leading to lysosomal storage disorders, LSDs, and glycogen storage disorders, lysosomal storage disorders, glycogen storage disorders. So these are all the inborn errors of metabolism. Okay, because of the deficiency of certain enzymes. All these disorders will come under autosomal recessive. All disorders will come under autosomal recessive, except, I have given you exceptions, except. Okay, except 2 disorders. Do you know what are these except 2 disorders? One is Fabry's disease. Okay, Fabry's disease, and the second disease is Hunter's. Okay, Hunter's disease. So Fabry's disease and Hunter's disease, these are the 2 exceptions. Okay, but all other inborn errors of metabolism, all other lysosomal storage disorders, all other glycogen storage disorders are autosomal recessive. All are autosomal recessive. I will show you. For example, if you look here, so here I am discussing. See, देखो, now Gaucher's, Krabs, Tay-Sachs, Fabry's. Okay, metachromatic leukodystrophy, Niemann-Pick's. Okay, on this side, Hurler syndrome, Hunter syndrome. Okay, now look, everyone is autosomal recessive. देखो, everyone is autosomal recessive. Exceptions are what? Fabry's disease. Okay, and also this side, everyone is autosomal recessive except Hunter. Okay, so except Hunter, this one, Hunter is autosomal recessive, sorry, X-linked recessive, and also Fabry's is also X-linked recessive. So now right here, this is Fabry's and Hunter's, they are X-linked recessive. Okay, they are X-linked recessive disorders. Okay, that's the one important MCQ which you should know. Next, usually autosomal recessive disorders. Now, in which people you see these autosomal recessive disorders? Do you have any idea? In which people they are most common, sir? In which people they are most common? These autosomal recessive disorders. In which people? Those people who are marrying within their relations. Okay, so those people who are marrying, like consanguineous marriages within the family, when they marriage, now they will get autosomal recessive disorders. Okay, consanguineous marriages. Okay, okay, consanguinity, consanguineous marriages. Okay, blood relation. If you marry a person who is related to you, very closely related to you, and when you have a baby, that baby is going to get one of the autosomal recessive disorders. Okay, more likely to get the autosomal recessive disorders. That's the one thing which you don't which you should never forget. So consanguineous marriages we can lead to autosomal recessive disorders. Okay, next, now let's discuss about some important points. First, let's discuss about a condition called as alkaptonuria. Okay, alkaptonuria. Alkaptonuria, in this condition mein, what is that enzyme deficiency? Alkaptonuria, sir. The patient, if whenever the patient goes to urine, the patient is going to have the mousy order urine. Okay, the mousy order. The urine is going to have the mousy order. Guys, can you tell me? Monica? Phenylketonuria. Sorry, first we are discussing about, first let me discuss about phenylketonuria, not alkaptonuria, phenylketonuria. Okay. Phenylketonuria, the patient is going to have the mousy order urine. What is phenylalanine? Alanine hydroxylase deficiency. Okay, phenylalanine hydroxylase deficiency. Now, okay, alkaptonuria. So the patient is having alkaptonuria. What is the enzyme deficiency? Homogenesic. Excellent, Monica. Yes, you are true. Okay. Yes, homogenesic. Okay, acid. Okay, homogenesic acid oxidase deficiency. So homogenesic acid oxidase deficiency can lead to alkaptonuria. Now, in this condition, tell me. So the homogenesic acid oxidase is deficient. So the homogenesic oxidase is not getting broken down. Now this homogenesic acid, it will start to deposit. It will start to deposit in cartilages. Okay, this homogenesic acid is going to start to deposit in the cartilages. So whenever you see the cartilaginous material, now it will look black in color. See, look here, this little blackish in color, little light bluish to blackish in color. So this condition is called as deposition of this homogenesic acid in the cartilaginous part, which is giving the black color appearance, is called as achronosis. Okay. Okay, no achronosis. Now this is an image based question that will come in your exam. Sir, achronosis is seen in alkaptonuria, homogenesic acid oxidase deficiency. Now what kind of disorder is it? It is autosomal dominant or autosomal recessive? It's an autosomal recessive disorder. Okay, it's an autosomal recessive disorder, sir. Okay, now in the beginning itself I have told you something. Sir, all the lysosomal storage disorders and all the glycogen storage disorders, they follow what? They follows autosomal recessive inheritance pattern. The first disease now I am going to discuss here, it's called as a Tay-Sachs disease. Tay-Sachs disease. Now in this Tay-Sachs disease, see, I find this beautiful images from the internet. Now in this Tay-Sachs disease, I am asking you, what is the enzyme deficiency? Can anyone tell me? So what is the enzyme deficiency? Hexosaminidase A, Tay-Sachs. Hexosaminidase A deficiency. Okay, hexosaminidase A deficiency. Now look here, in the Tay-Sachs disease, okay, what is happening? See, there is hexosaminidase A deficiency. Now normally hexosaminidase, it helps in the breakdown of GM2 gangliosides. See, there are some things called as a GM2 gangliosides. Okay, gangliosides, they are a type of lipids. Okay, they are a type of lipids. Okay, they are a type of glycolipids. Now these GM2 gangliosides need to be broken down. Who break down these GM2 gangliosides? So the GM2 gangliosides need to be broken down with the help of hexosaminidase A. If the hexosaminidase A, if it is deficient, do you think that GM2 ganglioside is going to be converted? It is going to be broken down? No. So GM2 ganglioside will start to accumulate inside the lysosomes. Now let me show you. Okay, for example, look here guys. Now here I am talking about the lysosomal storage disorders. Okay, lysosomal storage disorders. Dekho, GM2 ganglioside. Let's begin with the GM2 ganglioside. Now the GM2 ganglioside need to be converted, need to be broken down into GM3. Okay, with the help of which enzyme? With the help of this first enzyme, that is hexosaminidase A. When this hexosaminidase A, when it is deficient, it is not there. It is going to cause which disease? Tay-Sachs disease. So in Tay-Sachs disease, which levels are going to be elevated? GM2 ganglioside levels are elevated. Where? In lysosome. So in the lysosome, the GM2 ganglioside levels are elevated. The GM2 gangliosidosis. Now gangliosides are getting accumulated inside the lysosome, right? So now these patients, they go. So these are the lysosomes. Okay, these are the lysosomes. See, the lysosomes within the neurons, especially within the neurons, inside these lysosomes, who is getting accumulated? Sir, the GM2 gangliosides are getting accumulated. Okay, see the swollen gangliosides. Okay, the swollen gangliosides inside the lysosomes. So now in the lysosome, the gangliosides are getting accumulated. That is giving this onion skinning appearance. Okay, onion skinning appearance. Onion skin lysosome. Okay, guys, do you understand? Accumulation of GM2. That's a very important MCQ. If I am the examiner, I will give you. In which of the following lysosomal storage disorder, the GM2 gangliosides will start to accumulate inside the lysosomes? It is Tay-Sachs, hexosaminidase A deficiency. Because of the accumulation of this GM2 gangliosides in the lysosomes, you are going to have onion skinning appearance. Okay, something like this. Okay, onion skinning appearance is going to be seen inside the lysosomes. So they will ask you, what is this organelle, sir? This organelle is a lysosome. Who is getting accumulated inside? GM2 ganglioside. Okay, where? Especially in the neurons. It's a neurodegenerative disease. See, there will be progressive neurodegeneration in this disease. Okay, mainly the neural related problems are going to be seen, sir. Apart from that, in this disease, a Tay-Sachs disease, now can you guys tell me what is this? What is this that you are able to appreciate here in this Tay-Sachs disease? Come on, Monica, what is that? Excellent, Monica, excellent. Seems like you are a good girl. Okay, Monica, this is a cherry red spot. Okay, cherry red spot. Monica, can you tell me, sir, cherry red spot? Okay, cherry red spot in the macula. Excellent. One more, cherry red spot is seen in the macula, in the region of macula. Or they can say cherry red spot in the fovea also. Fovea, the macula. Okay. Now, my question to you is, right, cherry red spot is seen. Do you know in any other condition the cherry red spots are seen? So cherry red spots are seen in any other condition? Do you have any idea? Cherry red spots are seen. Do you think only cherry red spots are going to be seen in this hexosaminidase A deficiency Tay-Sachs? Any other area? No. You need to know the differential diagnosis of cherry red spot. So they go, look here, this is not something specific. Cherry red spots on the retina, they are not something specific, sir. Important MCQ for your exams. You will study this in your aphthal. Same. This is very important. Okay, from the ophthalmology point of view, this is very important. Which is something called as central retinal artery occlusion, CRAO. Okay, central retinal artery occlusion. Okay, Tay-Sachs disease, Niemann-Pick's, Gaucher's disease, and trauma. Okay, so in all these conditions, okay, central retinal artery occlusion, Tay-Sachs disease, Niemann-Pick's disease, Gaucher's disease, in all these conditions, you are going to see cherry red spot. Okay, you are all going to see the cherry red spot, sir. Okay, cherry red spot in the macula. Okay, now what is this? What are you looking at here? So these are the neurons which are now becoming obese. Now these are ballooned out neurons, sir. These are the ballooned out neurons. The neurons are becoming ballooned out because of the accumulation of the GM2 gangliosides. Now they will pull the water. Now they are getting ballooned. So this is called as ballooning of the neurons. This is cherry red spot. And don't forget, cherry red spot is not only just seen in this one condition, that is this disorder, Tay-Sachs, it is also seen in Niemann-Pick's as well as also Gaucher, and also seen in the trauma conditions. Trauma condition leading to edema. Okay, Berlin's edema. Now, what else you should know? What else you should know regarding this Tay-Sachs disease? My question to you, just answer me. Sir, in Tay-Sachs disease, is the patient is going to have hepatosplenomegaly? In Tay-Sachs disease, in this lysosomal storage disorder, very important MCQ, does the patient is going to have hepatosplenomegaly? Any idea? See, dekho. Look here. You know already we have discussed about the Tay-Sachs. Tay-Sachs, autosomal recessive. Sir, see, there is hexosaminidase A deficiency, so that GM2 gangliocytes levels are going to be elevated. Now, see, you are going to have cherry red spot. Okay, in the macula, macula shows the cherry red spot, and there is neurodegeneration. Okay, progressive neurodegeneration. So there is onion skinning of the lysosomes is seen. Okay, sir, cool. But here, have I ever mentioned that there is hepatosplenomegaly? So there is no hepatosplenomegaly. Okay, whenever you see the word hepatosplenomegaly, forget about the Tay-Sachs. So write no hepatosplenomegaly. Okay, hepatosplenomegaly. No hepatosplenomegaly. It is seen in other conditions. Now, let's discuss about the Niemann-Pick disease. Niemann-Pick's. Now, first question. Sir, in Niemann-Pick's disease, what is the enzyme deficiency? Okay, in Niemann-Pick disease, what is the enzyme deficiency? Any idea, guys? Yeah. Any idea Niemann-Pick's disease? Don't see, don't look. I'm asking you, Niemann-Pick disease, what is the enzyme deficiency? Look here, come to this table. Same. Look here, Niemann-Pick's disease. Normally the sphingomyelin, okay, the sphingomyelin, phospholipids, it's a phospholipid. Now the sphingomyelin need to be converted into ceramide. Okay, the sphingomyelin need to be converted into ceramide. Where? In the lysosomes. With the help of which enzyme? The sixth enzyme, sir, which is sphingomyelinase. Okay, so now deficiency of this enzyme, sphingomyelinase, will lead to what? Elevated levels of sphingomyelin. The sphingomyelin levels are going to be elevated in the lysosomes. Now sphingomyelin start to accumulate. So now the sphingomyelin is starting to accumulate inside the lysosomes. Okay, so that will lead to this kind of appearance. So what is this appearance, sir? This appearance, these are called as the zebra bodies. Okay, these are called as the zebra bodies. And zebra, you see the white black stripes, right? So these are the zebra bodies. Okay, so important points which you should know is that these lysosomal storage disorders are mainly seen in the Jewish population. Okay, the Ashkenazi Jews. Okay, so especially when you are giving your board exams, the USMLE kind of exams or the PLAB exams, this is very much important. So they will say something like there is this one person who is an Ashkenazi Jew, so coming from so on so, like, you know, Middle Eastern country. Now he is having so on so features. They will give you. Okay, so Ashkenazi Jews, important. They are seen in Ashkenazi Jewish population. It's an autosomal recessive disorder. Okay, autosomal recessive disorder. Now, what is the deficiency? Deficiency of an enzyme called as sphingomyelinase. Accumulation of what? Accumulation of sphingomyelin. Accumulation of sphingomyelin. Now, see, even in this Niemann-Pick's, the patient is having neurodegeneration. True, the patient is going to have neurodegeneration. The patient is going to have cherry red spots, but hepatosplenomegaly is seen in this condition. So there is hepatosplenomegaly. Okay, sir. So in this, the patient is going to have hepatosplenomegaly. And not only that, these patients are going to have the foam cells. MCQ. These patients are going to have the foam cells. What are the foam cells? Okay. So what are the foam cells? Foam cells are nothing but the lipid-laden macrophages. On light microscopy, you can see the lipid-laden macrophages. So those are called the foam cells. And you already know, I have discussed, on the electron microscopy, you can see the zebra bodies. Okay, you can see the zebra bodies. But one point I want you to know, sir, see, if I ask you something like this, zebra bodies are seen in. Now you can say, sir, zebra bodies are seen in this Niemann-Pick's. But zebra bodies are only seen in Niemann-Pick disease? No. Zebra bodies are not only seen in the Niemann-Pick disease. Let me show you. First, look here. See, zebra bodies and foam cells. Okay, zebra bodies and foam cells seen in Niemann-Pick's. Okay. Deficiency of sphingomyelinase. Sphingomyelin will start to accumulate. The patient is going to have cherry red spot, neurodegeneration, hepatosplenomegaly, foam cells and zebra bodies. Okay, sir, good. Looking everything good. But my question is, sir, these zebra bodies, are they only seen in this? No, sir. Look here. Zebra bodies. So where else you will see the zebra bodies? Fabry's MCQ. Okay, Fabry's. Fabry's also shows zebra bodies. But what is something important about the Fabry's? Sir, Fabry's is a lysosomal storage disorder, but it is having X-linked recessive pattern of inheritance. It is not autosomal recessive, it is X-linked recessive pattern of inheritance. Now, where else you will see the zebra bodies, sir? In any other place you will see the zebra bodies? Yes. You can still see here, dekho. Sir, zebra bodies are also seen in Hurler and Hunter. Okay, Hurler and Hunter. So now, right, zebra bodies are not specific. Okay, zebra bodies, especially for the PG exams, right? Zebra bodies are seen in Niemann-Pick's disease, Fabry's disease, Fabry's disease, Hurler and Hunter. Okay, Hurler and Hunter. So this is very important MCQ. Zebra bodies are seen in Niemann-Pick's disease, Fabry's disease, Hurler disease as well as Hunter disease. Now, next question. Sir, in this condition, Niemann-Pick disease. Okay, so you can ask me, sir, why we are discussing in detail, because 100% there is no doubt. In every damn exam, doesn't matter whether it is FMGE exam, NEET PG exam, INICET exam or board exams, 100% there will be questions on lysosomal storage disorders. Pakka, if it is even Central Institute exam, like INICET, many questions will come on the lysosomal storage disorders. Okay. Now look here. Now tell me, sir, hepatosplenomegaly is seen in Tay-Sachs? No. In Tay-Sachs there is no hepatosplenomegaly. Where hepatosplenomegaly is seen? Sir, hepatosplenomegaly, it is seen in Niemann-Pick's. But in which other lysosomal storage disorders you will see hepatosplenomegaly? Dekho, starting one, that is Gaucher's. In Gaucher's also you will see hepatosplenomegaly. Okay, done, sir. Next, where else, sir? In Hurler and Hunter. So in Hurler and Hunter also there is hepatosplenomegaly. So now, right, hepatosplenomegaly is seen in. Okay, hepatosplenomegaly, it is seen in Niemann-Pick's disease. True. Gaucher's. Next, Hurler and Hunter. Okay, so this is how you have to prepare for your exam. Okay, so whenever you are seeing hepatosplenomegaly, that can be seen. They can give something, a question like this: which of the following from the exam I will give something like this. Which of the following lysosomal storage disorders are shown to have hepatosplenomegaly? Niemann-Pick disease, Gaucher's disease, Hurler and Hunter. Which of the following lysosomal storage diseases are shown to have cherry red spot? Cherry red spots are seen in these three diseases. Okay, cherry red spots are seen in Tay-Sachs disease, Niemann-Pick's disease as well as the Gaucher's disease. Okay, 3. So done, sir. Now, after this, Gaucher's disease. Now, Gaucher's disease, can anyone tell me it is due to deficiency of? Any student, any intelligent student. Now, Gaucher's disease is due to deficiency of? Haven't studied biochemistry? Beta glucocerebrosidase. Okay, now look here. We are discussing about what? We are discussing about the Gaucher's, right? See, here it's the Gaucher's. So Gaucher's disease, normally the glucocerebroside, okay, the glucocerebroside need to be converted into ceramide. Okay, now who will convert this glucocerebroside into ceramide? Glucocerebrosidase. Okay, the third one, see, glucocerebrosidase. Okay, the beta glucocerebrosidase, or simply you can call it as beta glucosidase. Beta glucocerebrosidase. Okay, Indian books we simply say beta glucocerebrosidase deficiency is going to cause Gaucher's. Now, who is going to be elevated? Who is going to be accumulated in the body? In the lysosomes, the glucocerebroside is going to be accumulated. Okay, so what is that important MCQ that came in the recent NEET PG exam? In the recent NEET PG exam, this question was asked. Okay, this is the image-based question was asked. So what is this image-based question? So these are the macrophages, sir. These are the macrophages which are called as the Gaucher cells. Now, whenever you look at these macrophages, now there looks like a tissue paper which is crumpled. You take a tissue paper, you just crumple it. Okay, you just fold it into just do it like this. Now, when you open it, how it will look? Okay, how it will look? So that is something called as that crumpled tissue paper appearance. Okay, yes, crumpled paper. Okay, crumpled tissue paper appearance. So this is seen in Gaucher's disease due to deficiency of what? Deficiency of beta glucocerebrosidase. Okay, beta glucocerebrosidase. Now my question to you, sir, in this condition, Gaucher's, what are the two important clinical features which you should know? Okay, sir, Gaucher's is going to cause mainly bone related problems. Okay, bone problems, sir. Bone pain is going to be there. Okay, Gaucher's, the pain mainly the bone related pain, and these patients are going to have hepatosplenomegaly. Hepatosplenomegaly is present. We have discussed hepatosplenomegaly is seen in the Gaucher's. Okay, look here. In the Gaucher's, you see, Gaucher's, the patient is going to have hepatosplenomegaly. Now look, sir, one by one. Gaucher's disease, autosomal recessive, you know it. Deficiency of beta glucocerebrosidase. So glucocerebrosid levels are going to be elevated. The patient is going to have hepatosplenomegaly. The bone crisis, bone pains. Okay, bone pains. Very, very important MCQ. Avascular necrosis. Avascular necrosis of the femur. Okay, avascular necrosis of the femur. Bone crisis or bone pains. Okay, and you know the Gaucher cells will show crumpled tissue paper appearance. Okay, crumpled tissue paper appearance. Done, okay? So these are done with the Gaucher's disease. If you know this much, Gaucher disease is enough. Bone pain, hepatosplenomegaly, avascular necrosis, beta glucocerebrosidase deficiency. So cerebroside levels, okay, glucocerebroside levels are going to be elevated. So that will cause Gaucher cells. You will see the Gaucher cells where there is tissue crumpled tissue paper appearance. Now, done, sir. Autosomal dominant diseases, autosomal recessive disorders. Important points for your exams are completed. Now, let's discuss about X-linked recessive disorders. Okay. Now, shall we discuss about X-linked recessive disorders? We'll complete the topic, this entire topic, X-linked recessive disorders and X-linked dominant disorders. You can complete in hardly 20 minutes. Hardly 20 minutes. Shall we, guys? Shall we? Are you guys ready? Okay, done. Now, tell me, sir, mainly in these X-linked recessive disorders, which group is going to be affected? Do you know? Which group is going to be affected? Males or females? Which sexes are going to be affected in X-linked recessive disorders? Males or females? What do you think, guys? What do you think? Males. All problem for males. Males are mainly affected. Okay, in X-linked recessive disorders, the males are going to be mainly affected. Let me show you the example. Sir, now let's take X and Y. Now we are not discussing about the autosomes. Autosomes wala business completed. We are not discussing about the autosomes. We are discussing about now sex chromosomes, X and Y. They are the sex chromosomes. Now X and Y. Now it's a male. That's the father and mother, sir. X and X. Now these are X-linked recessive disorders, right? So I am taking now small x. Small x means affected. Mother is affected. Okay, the mother is affected. Okay, now whenever this couple, father and mother, okay, whenever this couple, when they have children, what are the possibilities? Dekho. Now x, small x. Okay, one capital X, one small x. See now mother, see now she is a carrier. Now these are recessive disorders, right? Now these are the recessive disorders. Now see, now she is having one small x, one capital X. Now she is a carrier, sir. Now she is not affected. She is just a carrier. And the father is healthy father. Now one female born XX. Now again one capital X, one small x. Again carrier female. Again carrier female. Now see, dekho, small x and Y, sir. And one capital X and Y. See, now tell me. See, this is a healthy. See, this is a healthy son. X and Y, healthy son. This is a carrier daughter, right? This is carrier. Carrier female. Now the carrier daughter. This is also carrier. Carrier female. Now who is affected at the end of the day? Here, who is affected is this male, sir. This guy, see, he is having a small x. Now he is only having one x, sir, one x. Females are having two x's, but male is having one x, and that too, this is a small x. So definitely Now in this heterozygous state, now it will express. Actually, I shouldn't say heterozygous here. Now there is only one X, right? So now the small x is going to show the disease. So this is affected son, affected son. This is normal son. So it's the males are going to be affected in X-linked recessive disorders. Males are going to be affected. Done. Now exceptions. Okay, let me write here the exceptions. Okay, exceptions. So in which conditions females are going to be affected? Female affected with X-linked recessive disorder. So in which condition the females are going to be affected with X-linked recessive disorders? Now first condition is Turner's female. Now guys, can you tell me what is a Turner female? What is a Turner female? So what is a Turner female? What is the problem with the Turner female? 45 X0. Now which means she is only having one X chromosome. Normally how she was supposed to be? How she was supposed to be? See, she was supposed to be something like this, XX. She was supposed to have two X chromosomes. Now she is only having one X chromosome. Now for example, now this female is having small x, 45 small x O. Now tell me that there is no capital X, there is no dominant genes, the dominant genes are not there now. Now she is having her time bad, normal X chromosome is gone. Okay, normally if a female is like this, for example, see dekho, now like this sir, I am telling you one female who is having this kind of genotype, capital X and one small x. Now this is an autosomal recessive disorder. Now is she going to have any problem? There is no problem, she will become the carrier, there is no disease. But now what if in Turner syndrome? In Turner syndrome, this is gone sir, capital X is gone. What if the capital X is gone? Now she is only having small x, X0. Now she will start to manifest the X-linked recessive disorders. So X-linked recessive disorders can be seen in Turner female. True. Okay, true. Second thing, lionization. Okay, so due to the process of lionization, yes, there is a chance that a female can have X-linked recessive disorders. Now we will get it out. Sir, what is this lionization? Lionization principle, Lyon's hypothesis is, now tell me, male. Okay, now just let's see here. Now males are having XY chromosome. Okay, X chromosome and Y chromosome. Okay, so male is having only one X, one set of genes. Now females are going to have XX. Now she is having two X chromosomes. So we need to balance, right? So what happens is generally in the body, one X chromosome is inactivated randomly in different cells. For example, randomly this X chromosome, one X chromosome is going to be inactivated. Okay, one X chromosome is going to be inactivated. Okay, so this is something called as a lionization principle. Now let's see, let's take one female. Now this female is capital X and small x, capital X and small x. Now time bad, her capital X chromosome have undergone inactivation. Now this capital X undergone inactivation. This is inactivated. Now who is going to be expressed now? Only this will be expressed. So the defective genes are going to become expressed. Now she will develop X-linked recessive disorders. Okay, now she is going to have the X-linked recessive disorders. So this is one possibility where a female, say now she is a female, now she is developing X-linked recessive disorder because of the process of lionization. Now in this lionization, what is happening? The capital X, the good chromosome is getting inactivated. Okay. Now the third possibility. Okay, the third possibility is now a deceased male cell. Now the deceased male. Let me write here. These are X-linked recessive, right? So deceased male, small x and y. Okay, this is right. Small x and y. So deceased male. Now he is marrying a carrier female. Okay, he is marrying carrier female, not marrying, they are having kids. You not to be married, right? So XY, now they are having kids with carrier female. So carrier female means like this, small x and one capital X, small x and one capital X. Okay, now देखो, Now if they having children, what are the possibilities? The possibilities will be something like this. Dekho. Small x, small x, small x, capital X. Okay. Y and small x and y. Next capital X and y. Okay, capital X and y. Now what you are looking at here? Okay, what you are looking at here, sir? Dekho guys. Now I told you general principle is what? Sir, X-linked recessive disorders hai na? These X-linked recessive disorders, they are going to mainly affect the males. Females are usually not affected. But now see in this combination, who is affected? Female, small x, small x. Now she is in homozygous state. Now female is affected. Okay, so never forget, when a deceased male, okay, deceased male with which disorder? With some autosomal recessive, with some X-linked recessive disorder, he is affected. Now this deceased male, when he married a carrier female, now they are going to have a female child. Now they are going to have a female child who is going to be affected. Okay, this is how females can be affected, sir. So now tell me, in which three conditions the female can get X-linked recessive disorders? One is in Turner syndrome. Second one is due to lionization. If one X chromosome is now inactivated, the healthy X chromosome is inactivated. Third condition is when a deceased male marries a carrier female. Okay, done, sir. Now examples that I should teach you. Okay, examples. What are the diseases? Diseases, sir. Now it's very simple guys. Diseases. Mnemonic is A, B, C, D, E, F, something like this. Okay, it will go on like this. Now, what are these diseases? A for, any idea? Hemophilia A and B. Hemophilia A and B are X-linked recessive disorders. Then hemophilia C. Hemophilia C, remember, very important, hemophilia C, it is autosomal recessive, right? In autosomal recessive part, you just add it, okay? So, autosomal recessive is hemophilia C, but hemophilia A and B are X-linked recessive disorders, okay? B for Burton's, A, gamma globinemia. Burton's A gamma globinemia, sir. Now imagine I am the person, okay, you should know this. If I am the person who is having Burton's A gamma globinemia, I am the one who is suffering with this. Okay, it's an X-linked recessive disorder. Okay, cool, sir. Now can you tell me what is the problem in Burton's A gamma globinemia? What is the problem, sir? Do you know what is the problem? Main problem, guys. Do you have any idea? It is defect in opsonization. Okay, defect in the process of opsonization. See, A gamma globinemia, the globulins, gamma globulins, antibodies are not getting produced. The B cells are not producing the antibodies. So, opsonization cannot happen. Very important. Opsonization defect is seen in Burton's A gamma globinemia. C for color blindness. Okay, C for color blindness. Which color blindness? Color blindness. Okay, which color blindness? It is red-green. Red-green color blindness. So this is a question which was recently asked. Red-green color blindness is most commonly seen in which sex? Males or females? It's X-linked recessive disorder, so males will be most commonly affected. So males are affected, sir. Okay. So color blindness. Okay. Not only this, one more disease which is called as a CGD. Okay, CGD. Monica, can you tell me? Monica, Kashyap, fun mode. Guys, can you tell me what is the CGD disease? We have discussed the topic of inflammation. What is CGD? Chronic granulomatous disease. Okay, chronic granulomatous disease. Okay, chronic granulomatous disease. Recently, in one of the exams, this question was asked. Nitro-blue tetrazolium test is done for detecting. Nitro-blue tetrazolium test. Nitro-blue tetrazolium test is done for chronic granulomatous disease. Okay. But anyway, that we'll discuss later. So color blindness completed as well as chronic granular blindness disease completed. D4, any idea? D4, let me show you. See this disease. In this disease, sir, the way the baby cannot stand properly. Okay, so he is now standing with the help of his upper extremities. And if you look at the calf of this baby, if you look at the calf muscles of this baby, the calf muscles are going to be very big, very big. That is called as a pseudo calf hypertrophy. Guys, can you tell me what is this? Duchenne's, excellent. DMD, Duchenne's. Duchenne's muscular dystrophy. Duchenne's muscular dystrophy, sir. So D. So Duchenne's muscular dystrophy. What is the problem in Duchenne's muscular dystrophy? So Duchenne's muscular dystrophy is X-linked recessive disorder. Okay, X-linked recessive disorder. Okay. Now what is the problem? Dystrophin protein is completely absent. Completely absent. Not just defect, not just deficiency, complete absence of the dystrophin. For example, if there is dystrophin deficiency, a defect, sir, that is going to cause Becker's muscular dystrophy. Becker's muscular dystrophy. So D stands for here Duchenne's muscular dystrophy as well as Becker's muscular dystrophy. Right? I am just writing here both things here itself. So Duchenne's muscular dystrophy and Becker's muscular dystrophy. Becker's is because of the deficiency. Duchenne's is because of the complete absence. Complete absence, sir. Now, because of that deficiency in that protein dystrophin, the patient is going to have weak muscles, weak skeletal muscles. So the sign is called as Gower sign. Gower's sign. Okay, the patient is like just standing with the help of this, the upper extremities, the Gower sign. Okay, done, sir. Now E for what is this? E stands for A, B, C, D completed. Now E. Any idea, guys? Any idea? E stands for? E stands for the highly elastic joints. I used to remember something like that. Okay, highly elastic. Okay, that I will discuss later. E. I used to remember something like is leash. Leash-Nyhan syndrome. Okay, Leash-Nyhan syndrome. Okay, E. So this is one thing which you need to by heart. Okay, Leash-Nyhan syndrome. Now, guys, can you tell me what is the problem in the Leash-Nyhan syndrome? Which enzyme deficiency? Leash-Nyhan syndrome. Leash-Nyhan syndrome is due to deficiency of HGPRT. Okay, hypoxanthine phosphoriboftransferase deficiency. Now, because of this deficiency of hypoxanthine phosphoriboftransferase, the patient is going to have Leash-Nyhan syndrome. Recently, I have posted this even in my Instagram also. Now, see, look here. See, what is your diagnosis? Now, dekho. Sir, the patient, see, what is your diagnosis? The question will something come like this. The patient is suffering with gout. The patient is suffering with intellectual disability and there is self-mutilating behavior in a boy. There is self-mutilating behavior, sir, in a boy. Okay. So, why specifically I am talking about a boy here? Because X-linked recessive diseases going to affect the males. Okay. So, here what is the diagnosis? The diagnosis is that if a person is suffering with gout, intellectual disability and self-mutilating behavior, this patient is suffering with Leash-Nyhan syndrome, hypoxanthine phosphoriboftransferase deficiency. It's a X-linked recessive disorder. Done, sir. E completed. Leash, E completed. E. After this what we have to discuss? F. Do you know what is this F? F, sir. Any idea about the F, sir? Any idea? Fabry's. Okay, see, Fabry's is a lysosomal storage disorder, but still, what is the deficiency? What it is coming under? It's coming under X-linked recessive. Okay, and fragile X syndrome. Okay, see, even fragile X syndrome, it will be discussed in the X-linked dominant also, but it's mainly X-linked recessive. Okay, it shows two patterns. Fragile X syndrome will show autosomal dominant as well as autosomal recessive. It is more of autosomal recessive when compared to X-linked. It's more of X-linked recessive, not autosomal. X-linked recessive, not the. It's also, of course, it also follows the X-linked dominant pattern, but most commonly it is X-linked recessive pattern disease. Okay, EF completed, sir. After EF, G, right? So G stands for G6PD deficiency anemia. Okay, G6PD deficiency anemia. Okay, in this G6PD deficiency anemia, more number of free radicals are going to be generated inside the RBCs. Now what happens? So in the RBCs, are you able to appreciate this precipitated hemoglobin? So this precipitated hemoglobin, this is called as Heinz bodies. So Heinz bodies are seen in G6PD deficiency. Now what are the cells called as? This helmet shaped cells. Okay, these helmet shaped cells, sir. Okay, where the splenic macrophages, they have just take a bite. The macrophages just have a take a bite. So these are called as bite cells. Yes, excellent. These are called as bite cells. Bite cells. So bite cells and hinge bodies, they are seen in, okay, bite cells and hinge bodies are seen in G6PD deficiency anemia. Don't forget, for your exams, what you will see? Bite hinge bodies and bite cells. The hinge bodies as well as the bite cells. Okay, EFG completed. Next H. H stands for what? You already know. The hunters. Okay, hunters disease. Okay, in hunter disease. Okay, so with this, all the X-linked recessive disorders are completed. X-linked recessive disorders are completed. Now the last 5 minutes topic is the X-linked dominant disorders. Okay, just you need to know the examples. That will be more than enough. See, let me write X-linked dominant disorders. Now my question to you, sir, in this X-linked dominant disorders, who are going to be affected? Now, who are going to be affected? For example, dominant, right? X-linked dominant. Now let me take a father, sir. Okay, now I am just keeping this prime symbol, which means defect, dominant. There is some gene defect. Okay, X, Y. The father is affected. Because the father is a male, right? He's going to have only one X chromosome. If it is affected, that's it. Okay, so he's going to have, he's married with now some normal healthy female, sir. Normal healthy female. Now tell me, what are the combinations? So girls are going to be affected, or boy childs are going to be affected, or girl childs are going to be affected. Now when this couple are married, okay, now he is having a defective X chromosome, X-linked dominant. So now see, capital X prime dash X again X Y last X Y. Who are affected? Just tell me, sir. Dekho, these are the dominant things. These are the dominant diseases. One copy is enough. Dekho now, sir. Now this is a female. So this is also female. Now females are affected, sir. Females are affected. Why? Because these are the dominant genes. So both the females are affected. So affected fathers, MCQ. Now this is a father. Okay, now he is a father. Now, right, father is going to spread disease to whom? So fathers, affected fathers spreads disease to daughters. Okay, affected fathers spread the disease to daughters. But here males are lucky, males are spared. Okay, males are spared, sir. Why? Because in males, X chromosome is always coming from the mother. Okay, X chromosome is always coming from the mother. Here in this condition, mother is normal. Here the mother is normal, right? Yeah, father doesn't affect the sons. True. One more. Father doesn't affect the sons. Mother. Okay. So father is affecting the daughters. Now, for example, in this condition, now I am taking this daughter, sir. This daughter. Okay. She survived. Now she is having the disease. Now she is marrying. Okay. She got into the marriage. Now she is married. Sir, see, this is the daughter. XX. Now she married. Okay. Now what she is doing? Sir, she is marrying, sir. She is marrying. Whom she is marrying? She is marrying with one more. Now she is marrying with one more person, person who is X and Y. Okay. Now tell me what happens. Okay, this is the affected mother, right? Affected mother. Now this is normal father, healthy father. healthy father. Now, देखो what happens. This is affected mother. Now, when they have children, okay, X dash and X, X dash and Y, XX, XY. Okay, so who are affected, sir? Now, the mothers are passing the disease to both sons as well as the daughters. Now, in this combination, okay, now I have taken this female. Now I have taken this female. Now, affected female. Now, when she married, okay, now when she married with a healthy person, okay, say now how much percent of the offsprings are affected? Sir, 50% of the offsprings are affected. That is, both female is affected. Female, this is the male. Okay, both females and males are affected. Male child as well as female child is affected. So now take a note. Affected females pass, okay, affected females pass the disease to 50% of offspring. Okay, you should know this, both the things. See, first condition affected male. Sir, first condition is the affected male, sir. Father affected. Okay, that's affected male. Affected male is affecting only the daughters. Affected male is spreading the disease to only daughters. But affected mother, affected female is spreading the disease both to daughters as well as the sons. 50%. Out of these 4 children, 50% of the children are affected. So both male affected, female affected. So father only targets the females, mother targets both males and females. Okay. So these are the points which I want you to know. The examples, the last and final thing, examples of X-linked dominant diseases. First one is Alport's. Mothers are doing the justice. For a mother, all children are equal. Okay, 50% of the children, females will be affected, 50% of the males will be affected. Okay, so Alport syndrome. Okay, the kidney pathology which will cause nephritic syndrome. Okay, Alport syndrome. Next, Rett syndrome. Next, vitamin D resistant rickets. Okay, vitamin D resistant rickets, and finally incontinentia pigmenti. Okay, incontinentia pigmenti. So these are the 4 diseases which I want you to know. Okay, these are the 4 diseases which I want you to know. Alport syndrome. Okay, these are the 4 diseases: Alport syndrome, Rett syndrome, and vitamin D resistant rickets, incontinentia pigmenti. These are the diseases which will follow X-linked dominant pattern. Okay, X-linked dominant disorders. So now let's begin without any further late. The topic which I am going to discuss now is non-Mendelian. Okay, non-Mendelian inheritance. So what are these disorders which follows non-Mendelian inheritance? Which diseases? Normally Mendel gave certain laws. This is how the diseases are going to be inherited. Okay, those are the normal: autosomal dominant, autosomal recessive, we have seen. Okay, so most of the diseases follow Mendel laws of inheritance, but there are certain special diseases which do not obey the Mendel's laws. What are they? Now, very important for your exams. Number 1 is mitochondrial disorders. Okay, mitochondrial disorders. Next, trinucleotide. Repeat disorders. Next, concept of those disorders where there is genomic imprinting is involved. Imprinting and gonadal mosaicism. Gonadal mosaicism. So These are the 4 conditions. Okay, these are the 4 disorders which do not obey the Mendel's laws of inheritance. We'll discuss about them one by one. Okay, most important things we will discuss. Okay, now having said that, see, first mitochondrial disorders. First, let's begin with the mitochondrial disorders. Okay, now let's begin mitochondrial disorders. Now, first of all, here you need to understand, sir, what is the basis for this mitochondrial disorders? The basis for the mitochondrial disorders is, okay, the basis for the mitochondrial disorders is, see, you need to understand there are 2 sets of genes in our body. Okay, so the genes which are present in the nucleus, the chromosomes, and the genes which are present in the nucleus, majority of our genes are present in the nucleus. But do you know, even mitochondria in the cell, for example, let me put it this way. So if you see, this is the cell. Inside the cell there is a nucleus. Inside the nucleus there is DNA and the genes. But there is one more organelle in the cytoplasm which is the DNA. This is the DNA. Sorry, that's the mitochondria. Even mitochondria have its own genetic material. Even genes are present in the mitochondria. Okay. So if those genes can be undergoing mutation, there is a possibility that those genes can undergo mutation. They will lead to diseases. Possible. Now my question to you is, from where do you get this mitochondria? Okay, from where do you get this mitochondria? See, all the mitochondria which are present in your cells right now, okay, all the mitochondria, whatever are there in your cells, in your entire body, they are all derived from the mother. They are, you got all the mitochondria from the mother cell. Okay, see, from the father, you only got the genes, you only got the nucleus actually. Okay, let me put it this way. Now imagine, this is the ovum. Okay, this is the ovum, sir. Okay, now ovum with the nucleus. Here is the nucleus of the ovum. This is the ovum. Now here is the cytoplasm present. In the cytoplasm, mitochondria are also present. Mitochondria also present. Now sperm, dekho. Now imagine this is the sperm. Now when the sperm is fertilizing the ovum, okay, when the sperm is fertilizing the ovum, only the male pronucleus, the pronucleus is going to enter into the ovum. So father is not giving any mitochondria, father is not giving any cytoplasm. Okay, the cytoplasm, entire cytoplasm and entire mitochondria which are present in you right now, it came from the mother. So all the mitochondrial genes are also coming from the mother. Okay, this is the first basic point which I want you to know. Okay, first write mitochondrial genes. From where you got them? Derived from mother. Okay, purely maternal in origin, sir. Okay, father is not giving any mitochondria. Now, based on this, now I want you to write these 2 statements. Now, if a mother is affected, if a mother is affected, mother is having some mitochondrial disorder. I will tell you what is the disorder later. Now, mother is having some mitochondrial disorder, sir. Now, if she is having 4 children, how many of these children will get this disease? All 4 will get the disease because now she is having the disease. Now she is having mutated mitochondrial DNA. The same mitochondria are going to enter into the 4 children. So all the 4 children are going to get the disease. Okay. So now affected mother, right? Affected mother spreads disease to all offsprings. Okay, to all the offsprings. Affected mother. But for example, if a male is affected with these mitochondrial diseases, male affected cell, I will show you. Okay. If a male is affected, then the father, affected father. Now father is having some mitochondrial disease, some problem in the mitochondrial DNA, some mitochondrial disease. Okay. Now do you think he is going to spread the disease to the offspring? Is it possible? No, not possible. So affected father is not going to spread the disease. Okay, affected father do not spread the disease. Okay, do not spread the disease, sir. Okay, this is the point which I want you to know. Now, for example, see, now let's take this as a father. Okay, now father and mother. Father and mother in the pedigree. Here I am showing you mother is affected. Okay, mother is affected. Now, if this couple, if they have 4 children, tell me. Now, let's see. They are having 2 boys, 2 boy children and 2 girls. Okay, 2 daughters, 2 sons and 2 daughters. Now, how many of them will be affected? Sir, mother is having mitochondrial disease. If mother is having mitochondrial disease, now all these 4 offsprings are going to be affected. Okay, all these 4 offsprings are affected with the disease, sir. Which disease? I will discuss, don't worry. Now, let's say, now this daughter, now she married, okay, she married some other guy who is a healthy guy. Okay, now this son married some girl, healthy girl. Okay, now what happens? Dekho. Now what happens? See, now you should tell me, answer, try to answer yourself. Now they are having 3 children, 2 boys and 1 daughter. Now these people are also having same 2 boys, for example, 2 boys and 1 daughter. Now tell me, which children will be affected? See, here the father is affected, here the mother is affected. When the mother is affected, all the offspring will be affected. Okay, when the mother is affected, all the offspring will be affected. Okay, see here the father is affected with the mitochondrial disease, but the father is not contributing any mitochondrial DNA to the offsprings. He is not giving any mitochondrial DNA. So here all the children are healthy. So this is how the pedigree chart is going to come in your exam. Okay, look, mother affected, all children affected. Mother affected, all children affected. Then it is 100% mitochondrial disease, sir. Okay, mitochondrial disease. Now after this, what else I want you to know? Examples of mitochondrial disorders. Mitochondrial disorders. Now what are the examples? These are very important, especially for the Central Institute exams. Okay, especially for the PG exams, these mitochondrial disorders are very much important. Now let me write MELAS. MELAS. MELAS is the first mitochondrial. It's the most common, sir. It's the most common mitochondrial disorder. First itself I am telling you, see, in all these mitochondrial disorders, sir, mitochondria are the sources. It's the source of ATP. Mitochondria is the place where ATP is getting generated. It's a powerhouse of our cells. So we need ATP continuously, sir. Who needs ATP continuously? Especially our neurons. Our neurons need the ATP continuously. So in all these mitochondrial disorders, there will be neuropathy. Neuropathy. Okay, even the visions of our visual activity also continuously vision is happening, right? Electrical impulses are traveling. Okay, so even these people will have problems with the eyes. Ophthalmoplegia is going to be seen. Okay. Okay, ophthalmoplegia and even vision loss can be seen, which is irreversible in nature. Irreversible vision loss can also occur. Okay, but generally speaking, there will be involvement of the nervous system and there will be also involvement of the eyes because they continuously need the ATP. Now in these disorders, the mitochondria are affected, the ATP production is affected, oxidative phosphorylation is affected. So these patients will have neuropathies and ophthalmoplegias. Okay, now what exactly is MELA, sir, if you ask me? MELA means mitochondrial encephalopathy. See, encephalopathy, something involvement with the central nervous system. Mitochondrial encephalopathy, lactic acidosis. Okay, ME, mitochondrial encephalopathy, lactic acidosis. Okay, normal aerobic respiration is not going to be possible because now the mitochondria are affected. Normal aerobic respiration is not occurring. Anaerobic respiration, ATP is not synthesizing. So anaerobic way of glycolysis. Okay, so lactic acidosis, stroke-like episodes. Okay, so this is the full form of the MELAS. So these patients are going to have mitochondrial encephalopathy, lactic acidosis, stroke-like episodes. Now if you ask me, in one of the exams this question was asked, so these patients are going to have seizures, sir. These patients are going to have the seizures. Okay, so which seizures? The patients are going to have tonic-clonic seizures. Tonic-clonic seizures. Okay, the patients are going to have the tonic-clonic seizures and apart from that, these patients are also going to have muscle weakness. Okay, so this is something important. These patients are going to have the tonic-clonic seizures. Okay. Yes, guys, this is the first mitochondrial disorder. The second mitochondrial disorder which I want to discuss is CPEO. CPEO. So what exactly is CPEO? If you ask me, chronic progressive external ophthalmoplegia. Okay, chronic progressive external ophthalmoplegia. See, again, eye involvement. Okay. Now, this one more thing. The CPEO means chronic progressive external ophthalmoplegia, and also this patient is going to have bilateral ptosis. Okay, bilateral ptosis is going to be seen. Okay, with bilateral ptosis. So this is the second mitochondrial disorder. Now the third mitochondrial disorder which I want you to know is MERF. Okay, MERRF, MERF. Guys, can you tell me what exactly is MERF? What exactly is MERF? MERF stands for MERF. It's the third mitochondrial disorder. Can you guys tell me what exactly is MERF? MERF means myoclonic epilepsy. Okay, myoclonic epilepsy, red ragged fibers. Okay, myoclonic epilepsy with the red ragged fibers. Now, in one of the exam, okay, especially this was asked in the Central Institute exam, and also this is very much important for your board exams. So those students who are trying to prepare for the USMLE exams, this was the question asked. Sir, what exactly happens in this MERF is the patient, okay, the patient is having point mutation, sir. Point mutation. Okay, where point mutation? On 8344th base pair. Okay, this is not the question. Okay, pair in mitochondrial DNA. So in the mitochondrial DNA, at this base pair, okay, 8344th base pair, there is a point mutation. Now, because of this, the patient is going to have which features? So features, clinical features, the patient is going to have are generalized seizures. Okay, the patient is going to have generalized seizures. The patient is going to suffer with dementia, and also generalized seizures, dementia, and cerebellar ataxia. Okay, so these are the three clinical features which I want you to know. So MERF, the patient, the MERF means myoclonus epilepsy, red ragged fibers. Now, if you ask me, sir, what exactly are these red rag fibers? I will show you the image. Okay, I will show you the image. See here. Now, see, this is the muscle biopsy. Okay, here is the muscle biopsy. Now, in the muscle biopsy, you can very clearly see this red color appearance. Okay, so these are the mitochondria. Okay, these are the mitochondria, sir. Okay, so in the muscle biopsy, you can very clearly see. This is very important image based question. Now, look here. On electron microscopy, here, this is the electron microscopy, sir. Okay, this is the electron microscopy. Now, whatever you can see here, all these are the mitochondria, damaged cristae. In the mitochondria, you know, right, the cristae are present. The cristae, the inner mitochondrial membrane forms the cristae. So, the damaged cristae, it is forming this disc-like appearance. Okay, it's forming a disc-like appearance. So, this is called as the abnormal mitochondria with circular damaged cristae forms a concentric membranous ring-like structures. This is called as phonographic records. Okay, phonographic records, sir. You know that phonograph, right? Like, you know, we'll put the, phonographic disc to play the music. So, these are the phonographic records. Okay, so phonographic records are seen in which condition? So, they are seen in MERF, myoclonus epilepsy, red ragged fibers. And not only that, here you can see this mitochondria, inside the mitochondria, this cristae, they are arranged like a cars, car parking. Okay, car parking areas. Okay, so look here. See, you can see the thick parallel rods inclusions. This is something called as a parking lot appearance or parking lot inclusions. Okay, the parking lot inclusions in the damaged mitochondria. You can see these inclusion bodies. They are called as a parking lot appearance. So for your exam, especially for the Central Institute exams, you should know phonographic records. are seen. These red ragged fibers are nothing but the mitochondria, sir. These red ragged fibers, okay, whatever I am highlighting, this in the periphery of the muscle, these are the mitochondria. Okay, done. So after MRF, now let's go with the NARP. NARP. So I want you to answer. Come on, guys. Come on. What exactly is NARP? Any idea? NARP. NARP. NARP stands for, yes, neurogenic ataxia retinitis pigmentosa. Okay. Neurogenic ataxia. Okay, neurogenic ataxia retinitis pigmentosa. Next, mitochondrial disease. Not much important points about the NARP. Next mitochondrial disease is LON. What exactly is LON? Leber's hereditary optic neuropathy. So Leber's hereditary optic neuropathy. Now here important point which I want you to know is for the Central Institute exams. So here in this Leber's hereditary optic neuropathy, the complex 1 in the mitochondria, you know, right, the complex 1 which is involved in the formation, like, you know, of the ATP, complex 1, 2, 3, 4. So complex 1 is damaged, okay, because of the mitochondrial genes which are mutated. The complex 1 dysfunction is seen. There is a dysfunction of the complex 1, and even there is optic death. Okay, I should say the cellular death in the optic nerve, MCQ. Okay, cellular death in optic nerve, or they can say optic nerve atrophy. Okay, optic nerve atrophy. So in this condition especially the Leber's hereditary optic neuropathy, see in the name itself it's there is optic neuropathy. The patients are going to have acute, means sudden, acute or subacute vision loss. So just tell me one mitochondrial disorder which presents with irreversible vision loss, that is Leber's hereditary optic neuropathy, where there is complex 1 dysfunction, optic nerve is going to be damaged. There is cellular death in the optic nerve that will cause acute or subacute visual loss. Okay. Now last one which I want you to know is, okay, Leigh syndrome. Okay. Okay, Leigh syndrome, sir. Sir, what exactly happens in this Leigh syndrome? So the genes mutated, okay, Central Institute exams important, the genes mutated are SURF SURF. SURF1 gene is mutated in 80% of the cases. In 80% of the cases, the gene mutation is SURF1 gene mutation. In 20% of the cases, the gene mutation is MTATP6. Okay, MTATP6 gene mutation in 20% of the cases. Now, because of these gene mutations, again there is dysfunction of complex 1 of the mitochondria. Okay, again there is dysfunction of the complex 1 of the mitochondria. So in this condition, Leigh syndrome, what are the clinical features? Any idea, guys? What are the clinical features seen in this condition? Sir, Leigh syndrome, what are the main clinical features? The patient are going to have, yes, visual involvement will be there, definitely the central nervous system involvement will be there, and the muscle involvement is going to be there. Now, any eye abnormalities, can you tell me? Sir, this mitochondrial disorder, it's going to present with nystagmus, sir. Okay, nystagmus movement. Okay, nystagmus is going to be seen. True, one thing. And in this condition also, optic nerve atrophy. Okay, optic nerve atrophy is going to be seen. True. And in this condition, very important MCQ. Okay, these patients are going to have hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy is seen. So in which mitochondrial diseases hypertrophic cardiomyopathy is seen? See, go for the Leigh syndrome. Okay, here the patient is going to have the hypertrophic cardiomyopathy. Apart from this, the patient is going to have ataxia, and the patient is also going to have the neurological involvement, that is the psychomotor regression is going to be seen. Okay, regression. psychomotor regression, and these patients, it's very hard for them to survive. Okay, failure to thrive. Failure to thrive. So one important point which I want you to know here is this one, sir. So the MCQ point is hypertrophic cardiomyopathy. Okay, nystagmus. Nystagmus, hypertrophic cardiomyopathy, ataxia, psychomotor regression, failure to thrive. These are all the features of Leigh syndrome. The gene mutations are SERP1 gene mutation and MT-ATP6 gene mutation. The mutation in these two genes can lead to the dysfunction in the complex number 1. Complex number 1, sir. Okay, so with this, Leigh syndrome is also completed. So which syndromes we have discussed? MELAS, mitochondrial encephalopathy, lactic acidosis, stroke-like episodes. Okay, the patients are going to have the tonic-clonic seizures. In CPEO, chronic progressive external ophthalmoplegia with bilateral ptosis. MERF, very much important. Image-based questions are very much important, sir. Here, the muscle biopsy is showing the red ragged fibers, and electron microscopy is showing the phonographic records as well as the parking lot appearance. Parking lot appearance because of the inclusion bodies. And after this, what we have discussed, MERF completed. After this, NARP, neurogenic ataxia retinitis pigmentosa. LON means Leber's hereditary optic neuropathy, where there is sudden acute or subacute visual loss is going to be seen because of dysfunction in the complex number 1. And Leigh syndrome, SERP gene mutation as well as MT-ATP6 gene mutation, done, sir. Now after this, there are even other mitochondrial diseases present. Let me tell you one more. Pearson, okay, Pearson syndrome. Okay, so Pearson syndrome is also a mitochondrial disease, same mitochondrial disease. See, tell me, in Pearson's syndrome, the patient is going to have which involvement? The patient is going to have which involvement? See, I used to remember P for P. The patient is going to have pancreatic insufficiency. Okay, the patient is going to have the pancreatic insufficiency as well as, very important, the second thing, sideroblastic anemia. Okay, so pancreatic insufficiency as well as sideroblastic anemia. So just as a recap, okay, tell me, what are the important mitochondrial diseases? The important mitochondrial diseases are MELAS, CPO, MERF, NARP, Leber's hereditary optic neuropathy, Leigh syndrome, and Pearson's syndrome. Okay, Pearson's syndrome, sir. Okay, now after this, let's discuss about the mitochondrial diseases completed, sir. I have shown you the mitochondrial inheritance pattern also. If mothers are affected, all offsprings are affected. If the father is affected, no offspring is going to be affected. Now after this, the next topic which I want you to know is trinucleotide repeat disorders. Okay. Now, so do you know any trinucleotide repeat disorders? Normally in these disorders, what happens, sir? Simple. What happens, sir? The trinucleotide repeats in the DNA. The trinucleotide repeats, for example, CAA or CAG, CAG. So this is a trinucleotide, right? You know, 3 nucleotides together called as a trinucleotide. So these trinucleotides are going to repeat, okay, thousands and thousands of times. The same trinucleotide repeat is going to repeat again and again. Okay, normally it should be CAG, CAA, CGA, something like that, CTG, something like that. Okay, but now what happens in this trinucleotide repeat disorder, sir? For example, now imagine this is the DNA. Now in this DNA, now the same repeats are going to, like, you know, same trinucleotides are going to repeat. CAG, CAG. Again, one more, CAG. Cysteine, adenine, guanine. Again, cysteine, adenine, guanine. Cysteine, adenine, guanine. Normally, yes, these repeats are there in our DNA, but these more number of repeats are going to occur. Again and again. So that's going to cause a disease, sir. Okay. So now, first let's see what are the trinucleotide repeat disorders. There are 4 important trinucleotide repeat disorders. One is Huntington's. Okay, Huntington's disease. Next, Fragile X syndrome. Next, Friedreich ataxia. and last one is myotonic dystrophy. So these are the 4 disorders which are due to trinucleotide repeats, more number of trinucleotide repeats. Now in your exam, if you are going for your FMG exam, now this is the least which you should know. The least which you should know is, sir, in Huntington disease, yes, it's a trinucleotide repeat disorder. Which trinucleotides are going to repeat again and again? Remember, it is CAG repeats. Okay, the same CAG is going to repeat again and again. Sir, in Fragile X syndrome, what is the trinucleotide repeat that's going to occur again and again? It is CGG repeats. Okay, CGG, sir. Now in Friedreich ataxia, I used to remember something like this. Ataxia means what? Ataxia means gait abnormality, right? Gait abnormality. Gait. So gait is not proper. So here the repeats are GAA. Okay, GAA, gait, right? GAA. I used to gait, GAA. Right, gait. I used to remember something like that. So GAA repeats are going to occur again and again. Sir, myotonic dystrophy. I used to remember myotonic dystrophy. So tonic, T. So CTG. So everything. Okay. See, in most of the things it's starting with the same C, sir. C. Here also C. Everything starts with the C. And even ending with G. Easy, I have given you mnemonic. Sir, this one. Wait, yeah, this one. Ataxia, Friedreich ataxia. Ataxia means gait, GAA. But everything starts with C. Everything is going to end with the same G. So now tell me, Huntington's disease, what are the repeats going to occur? CAG repeats, Fragile X syndrome CGG, and Friedreich ataxia GAA, and the myotonic dystrophy it is CTG. These are the disorders. Now look here. First, let's discuss about the Fragile X syndrome. Okay, Fragile X syndrome. Okay, the Fragile X syndrome, sir. Now, look, this is your normal X chromosome. Now, if you look here in this Fragile X syndrome, look here, it's just looking like that part of this X chromosome, it is falling off. Okay, this part of X chromosome, it is getting separated, detached. It's very much a fragile area. So there is this fragile area. Why that fragile area? It's because of that area is going to be not stained, sir. That area is not going to take the stain. Okay, when you stain that chromosome, that particular part of the chromosome is not going to take a stain. So that it's looking like it's a fragile region. Okay, so Fragile X syndrome. Now tell me, in Fragile X syndrome, what are the important points which you need to know? Sir, what is the gene mutation? Gene mutation. It is FMR1 gene mutation. If it is a FMR1 gene mutation, FMR stands for familial mental retardation. So it's a familial mental retardation, one gene is going to be mutated, and you already know in this Fragile X syndrome, what are the repeats? What are the trinucleotide repeats that are going to occur? So it's in Fragile X syndrome, it is not CTG. Just wait. So in this Fragile X syndrome, it is CGG. Yeah. Okay. CGG repeats are going to occur. Now look here. So these are the patients. Okay, these are the patients who are suffering with the Fragile X syndrome. Now in this Fragile X syndrome, what are the important points which you need to know? Sir, okay, CGG repeats are there. Okay, how many repeats can actually cause the disease? Dekho, in you and me, in most of us, the CGG repeats less than 55 is considered as normal. Some books mention less than 45 is normal, but less than 55 repeats. If we have 55 repeats, that's okay. In you and me, we have the same repeats, sir. Same repeats. CGG repeats are going to be 55. That's okay. Now, more than 55 to 200. That's considered as a premutation. Okay. Premutation. Now, when you are having this premutation, then you are going to suffer with, guys, remember, then you are going to suffer with Fragile X tremors, not Fragile X syndrome. Then you are going to have Fragile X tremors. Okay, it's a pre-mutation, sir. Now if you have more than 200 repeats, then it will cause a fragile X syndrome. Then it's going to cause a fragile X syndrome. Now important points, guys, look here, these are the patients who are having the fragile X syndrome. Look at their face, how their face is, long face, long ears, long mandible, okay, long face, long ears, long mandible. But one important point especially for your exam is, if you look at their nose is normal. What is normal? N for N. Nose is going to be normal, but they are going to have elongated long face with long ears and long mandible, sir. Okay. Now apart from this, what else are the important points which you need to know regarding the fragile X syndrome? See, these patients post-pubertal means once they reach their puberty, their testicular size is going to increase very much. So there is a macroarchidism, okay, large testis, okay, which is called as a macroarchidism. Apart from this, what else? So these patients are going to have, CVS problem, sir. Which is CVS problem? So these patients are going to have mitral valve prolapse. Okay, what is the CVS problem? It's a mitral valve prolapse. So that these patients can also have the mid-systolic clicks, okay, murmur. Because of this mitral valve prolapse, that's a mid-systolic clicks. These patients are going to have, like, enlarged, okay, and the dental crowding will be seen. That's not that important. I am not going into little small details. No, not enough. It's not needed. So post-pubertal macroorganism is seen. See, as a CVS abnormality, mitral valve prolapse can be seen. Okay, cardiomegaly can be seen. Dilation of the aorta can be seen in these persons. But that's not important. Important point is, sir, I have shown you here the four fellows who are suffering with fragile X syndrome. All these four fellows are the males. Why? Why fragile X syndrome? All the persons who are getting affected are males. Because fragile X syndrome, remember, it is X-linked recessive disorder. It's an X-linked recessive disorder. Okay, sir, all these X-linked recessive disorders, okay, it's an X-linked recessive disorder, sir. All X-linked recessive disorders are going to occur mainly for the males. Then what are the females? Females are the carriers. Females usually do not affect with the fragile X syndrome. Females are carriers. And these females can also have problem. Do you know what is that? Important MCQ. So these females will have premature ovarian failure. Okay, premature ovarian failure, sir. Okay, so whenever you see a question with a large face, elongated face, large mandible, with large ears, but the nose size is normal, with the mitral valve prolapse, okay, mitral valve prolapse with the murmurs, you should think about this patient is having fragile X syndrome. And they can ask you in image based question also, okay, like they can give you X chromosome something like this with this with these ends which are like little getting detached from the chromosome. Okay, so these are the fragile X syndrome, which repeats, CGG repeats. More than how many number of repeats? More than 200 repeats. Okay, more than 200 repeats. In females, you can have premature ovarian failure. Now, after this, what else I want you to know is, let me write here, there is something called as Sherman's paradox. Sir, what exactly is this Sherman's paradox? This Sherman's paradox means, now the father is going to give this disease to the son. Imagine, a father is giving the disease to the son. Now this son is going to give the disease to his son. Now with generation to generation, this disease is spreading, right? Now, do you know what happened to the number of trinucleotide repeats? So, with every generation, right? With every generation, for example, imagine there is his father. Okay, father is giving the disease to his son. This father is again giving the disease to son. Now, for example, he is having how many repeats? For example, he is having 500 repeats in CGG. Now, in this fellow, in this son, he is going to have 600 repeats. Now, in his son, he is going to have 750 repeats. Means with every generation, right? With every generation, number of repeats increases and severity of the disease also increases. Okay, so severity also increases, and you can expect, for example, if the father have manifested the disease at 30 years of age, now you can expect, you can anticipate, okay, you can anticipate that in the son who is having the same disease, he will develop the disease by 25 years because the number of repeats are going to increase. The number of repeats are going to increase, the severity of the disease is going to increase, so that you can do anticipation. So, right, important MCQ, anticipation. Okay, anticipation is seen in the trinucleotide repeat disorders. Okay, with every generation the number of repeats increases, severity of the disease also increases. So, you can anticipate that if the father is having a disease by 30 years of age, he will also develop, the son is also going to develop the disease a little earlier, okay, by 27 years of age or 25 years of age. Okay, so that's the Sherman's paradox. Now, after this, let me show you, this is the Netter's image. Okay, this is the Netter's image. You need to identify what is this disease. So, the disease where, see, once the patient is having a grip, now when you give something into his hands, he is going to have a grip, but see, he is having a contraction, muscular contraction is possible, but the relaxation is not possible, sir. See, difficulty in releasing the grip, difficulty in releasing the grasp or the grip, sir. So, guys, can you tell me what is this disease? So, this is myotonic dystrophy. Okay, let me write here itself, myotonic dystrophy. Okay, myotonic dystrophy, sir. Now, in this myotonic dystrophy, what are the important points which you need to know for your exam? Okay, myotonic dystrophy. Sir, what are the repeats? Myotonic, remember, myotonic, it's a CTG. Okay, CTG repeats. The repeats are going to be CTG repeats. See, this myotonic dystrophy, it follows which inheritance pattern? Autosomal dominant inheritance pattern. It follows autosomal dominant inheritance pattern. Important points about this disease. So, these patients who are having this myotonic dystrophy, they have difficulty in releasing the grip or the grasp. Okay, difficulty in releasing the grasp. And if you look at their eyes, these patients are going to have the cataract. So, this is the cataract. It's looking like a Christmas tree. Okay, it's looking like a Christmas tree. Okay, lightened Christmas tree. So, these are called as, any idea? Christmas tree. Cataract. Okay, they are going to have the Christmas tree cataract. Apart from this, what else you should know? Sir, in this disorder, myotonic dystrophy, look at this person. Okay, yes, difficulty in grasp, okay. But if you look at this person, now he is having baldness, sir, male pattern baldness, very important. So, male pattern baldness are going to be seen, and these patients are going to have, okay, hatchet faces. Okay, these patients are going to have the hatchet faces, something like this. Something like this. Okay, hatchet faces is going to be seen. So, these are the two important points. Whenever you see the word hatchet faces, you need to think about myotonic dystrophy. Okay. And these patients are going to have the male pattern baldness. Okay. Now, very much important point. In which disease today we have discussed? The patient is having macroarchidism, post-pubertal macroarchidism is seen in. Just now we have discussed here. Dekho. Look here. See, large testes, macroarchidism, it is seen in Fragile X syndrome. But now in this myotonic dystrophy, the patient is going to have the testicular atrophy. MCQ. This is the MCQ, sir. Testicular atrophy. Okay, testicular atrophy is going to be seen, sir. Apart from this, any other important point for your exam? Yes, there is one important point for your exam, that is the patient is going to have the cardiomyopathies, arrhythmias. Cardiomyopathies with the heart blocks. Okay, so these are the important points which I want you to know. Sir, myotonic dystrophy, important points. See, autosomal dominant disorder, tonic, so CTG repeats are going to be seen. So difficulty, these patients are going to have the difficulty in releasing the grasp, contraction okay, but relaxation is not possible, difficulty in releasing the grasp. Christmas tree cataracts are going to be seen. The patients are going to have the Hatchet's faces, long elongated Hatchet's faces are going to be seen. Male pattern baldness, testicular atrophy is going to be seen with arrhythmias. The cardiovascular involvement is going to be the arrhythmias and conduction blocks. Cardiomyopathies, conduction blocks are going to be seen. And in these patients, the patient is also going to have the muscular atrophy, myotonic dystrophy, right? So muscular atrophy, important point. These patients are going to suffer with muscular atrophy and respiratory difficulty, because even the diaphragm is a muscle. It's also having a problem now. So there is respiratory difficulties. So all these are the important points which I want you to know. At least, but before going to your exam, remember this testicular atrophy, male pattern baldness, Hatchet's facies, Christmas tree cataract, and CTG repeats. Okay, everything is a question. I cannot say, like, I cannot give you the discounts here. Okay, CTG repeats important, autosomal dominant important, difficulties in releasing the grip. This is a very important keyword. Okay, so these are the points which I want you to know. Now, completed, sir. Trinucleotide repeat diseases, important points I have completed. Now, after this, let's discuss about the genomic imprinting. Genomic imprinting. So what exactly is this genomic imprinting, sir? What is the concept of genomic imprinting? Okay, let me tell you. Look here. First of all, guys, can you tell me what is this baby suffering from? There is this baby who is having a short stature and obese. Short stature and obesity. Okay. Now, do you know he is suffering with which genetic abnormality? I will tell you. Okay, I will tell you. But before going there, you need to understand what is genomic imprinting, sir. Now, let me take chromosome number 15. Okay, chromosome number 15. So 15 chromosomes, 215 chromosomes. So we all have the 215 chromosomes, right? One from the mother, one from the mother, other from the father. Father, mother. So I have two chromosome number 15s, one from the mother and one from the father. Now the first gene I want to discuss here is called as a SNRP gene. Okay, the gene which I want to discuss here is called as a SNRP gene. So this SNRP gene, where it is present? Sir, it is present both, for example, let's see, for understanding purpose I am keeping here. Okay, the SNRP gene is present both in mother as well as father. So this SNRP gene which is present in the mother, it is silenced. It's by methylation process. By doing the methylation, by adding the methyl group to these genes, it is completely silenced, sir. It is silenced, means it is sleeping. The SNRP gene which is present in the father, the SNRP gene which is present in the father, it is active. Means by default normally itself, the SNRP gene which is present on my maternal chromosome, it is totally silenced. Normally how it should be, according to Mendel, see, according to Mendel, there are two sets of genes, one from mother and one from the father. Two alleles, one from father and from the mother. Same gene is having two alleles, one is on the mother, one is on the father. Now, whoever is dominant, whoever the gene is dominant, okay, so that gene is going to be expressed. Whatever the gene is dominant, that gene is going to be expressed. Other gene is going to be not expressed. But in this condition, see, this is not following the Mendel's law. Yes, two genes are there, two SNRP genes are there. By default itself, all our maternal SNRP genes are silenced. So, which SNRP gene is active? only paternal side SNRP gene is active. Okay, one is silenced, one is active. Done. Now, let me tell you about the one more gene, sir. Okay, one more gene. Now, same 15 chromosome. This is the same 15 chromosome. One from the father and one from the mother. 15 chromosome from the father, 15 chromosome from the mother. Now, I am talking about a gene which is called as UBE3A gene. Okay, UBE3A gene. So, is it important for your exam? Yes, especially for the PG exam. This is very much important. SNRP gene, UBE3A gene. Now, same genes are present both on mother as well as father also. Same gene is present. But in the mother, this UBE3A gene, okay, it is active. It is active. Active means it is going to express. It is going to be expressed. But here, this UBE3A gene on the father, these genes are methylated. By doing this process of methylation, the genes are going to be totally silenced. So, gene silencing, how can you do? By doing methylation. By doing methylation, sir. Now, silenced. Okay, silenced. Now, what happens? This is something normal. What happens? Now, dekho, what happens in Klinefelter syndrome? Okay, what happens if there is some problem? Okay, not Klinefelter syndrome, sorry. What happens if there is any problem with this imprinting process? First, let's write the diseases here. The two diseases which I want to discuss here is Prader-Willi syndrome and Angelman. Okay, so Prader-Willi syndrome as well as the Angelman syndrome. So what is the problem in the Prader-Willi syndrome? Problem क्या है Prader-Willi syndrome में and Angelman syndrome? See, look here. Normally, the SNRP genes. So SNRP genes are active. Okay, SNRP genes are active in the paternal chromosome, but the SNRP genes are totally silenced in the maternal chromosome. Now, what happens if there is a deletion of this paternal chromosome? Paternal chromosome is gone, deletion. So now I am left with only maternal chromosome number 15. On maternal chromosome number 15, the SNRP genes are not active, they are silenced. So I completely lost the SNRP genes. Okay, so Prader-Willi syndrome, what is the problem? Let me write here. The problem in Prader-Willi syndrome, how can you get the Prader-Willi syndrome? Is one way. I am telling you what is the one way. It is paternal deletion, paternal chromosome number 15 deletion. So deletion of paternal chromosome. Or try to understand like this. There is one more possibility. Prader-Willi for P. See, remember simply Prader-Willi, paternal deletion of chromosome number 15. Now, one more possibility how you can get Prader-Willi syndrome is, imagine. Now, as a person, you are supposed to have, as a human, you are supposed to have 215 chromosomes. 215 chromosomes. One from the father as well as one from the father, one from the mother. Okay, one from the father and one from the mother. What if you got chromosome number 15, both the chromosome number 15 from the mother itself? From the mother, you got two 15 number chromosomes. Father is not donating the 15 number chromosome. This is called as uniparental disomy. Uniparental disomy, which means uniparental, from one parent. 215 chromosomes came from where? Mother. So maternal disomy. So maternal disomy of chromosome number 15. Now you already know, sir, if both the chromosomes come from the mother, 15 number chromosomes, in the mother, you know that the SNRP genes are completely silenced. The SNRP genes are completely silenced because of the methylation. So the SNRP genes are not going to be active in you. So that's going to cause which syndrome? That's going to cause the Prader-Willi syndrome. So now tell me, in your exams, these are the questions. So the Prader-Willi syndrome, is it due to paternal 15 deletion, or you can get the same Prader-Willi syndrome because of the maternal disomy 15. Maternal disomy 15. Now, then what is the Angelman syndrome, sir? In Angelman syndrome, what is the problem? Sir, in Angelman syndrome, the problem is, again, look here, UBE3A gene. UBE3A gene. So, this UBE3A genes, they are active only on the maternal chromosome number 15. Maternal chromosome number 15. What if you lost the maternal chromosome? Deletion of chromosome number 15 from maternal chromosome number 15. Now this UBA3A genes, yes, they are also present on the father side, but they are inactive. So you completely lost those genes. So in Angelman syndrome, what is the problem is a maternal 15 deletion. So deletion of chromosome number 15, which came from the mother. So maternal 15 chromosome is lost. So then it's going to cause Angelman syndrome. Or you can get the Angelman syndrome. How you will get the Angelman syndrome? If you receive chromosome number 15, 2 copies of chromosome number 15, both from the father. Okay, so that is paternal disomy. Paternal disomy 15. If you get both the 15 number chromosomes from the father, then you do not have functional UBE3A genes. They are not there in your body. So then you are going to get the Angelman syndrome. So the important points I have give you about the Prader-Willi syndrome and Angelman syndrome. Prader-Willi, Prader, P for P, paternal deletion of chromosome number 15. Angelman syndrome is the maternal, angel is the mother. Deletion of chromosome number 15 from the mother is going to cause the Angelman. And one more possibility also I explained you, that uniparental disomies can also lead to the Prader-Willi as well as Angelmans. Now in Prader-Willi, dekho, sir, in Prader-Willi syndrome, let me write here important points about the Prader-Willi. In Prader-Willi syndrome, what are the clinical features, sir? See, this is the guy who is suffering with the Prader-Willi syndrome. Now what are the clinical features are going to be seen? First clinical feature number 1, so Prader-Willi syndrome clinical features, short stature. Okay, short stature. Second, obesity. Okay, obesity is going to be seen. So these patients are going to have hyperphagia, excessive, they can't control, their eating habits. There will be hyperphagia. Hyperphagia is going to be seen. They are going to have hypotonia in the muscles. Okay, the muscles are going to show the hypotonia. Their body skin color is also going to be very much fair. Hypopigmentation. Okay, 3H, H. Okay, there is a mnemonic that is H30. Okay, H30. Okay, H30 is the mnemonic, sir. H30. H for H3, 3 H's, sir. These patients are going to have the hyperphagia, hypotonia as well as hypopigmentation. O for obesity. These patients are going to have obesity, short stature and obesity. Now in these patients, there is, why obesity means they have high levels of ghrelin. Okay. Sir, the ghrelin levels, it causes the hunger, sir. Okay, it causes the hunger. So these patients are going to have a low impulse control. They cannot control their eating. They will be doing hyperphagia, sir. Low impulse control. Very important MCQ. Low impulse control. Okay, so they cannot control their food habits. Okay, so that's a low impulse control because of the high levels of ghrelin hormone. Ghrelin increases the hunger. Normally in you and me, ghrelin is produced during fasting states by the pancreas. Okay, pancreas, the epsilon cells of the pancreas are going to produce a ghrelin. Ghrelin increases the hunger, we know it. But in which genetic disorder, the ghrelin levels are going to be elevated. The patient is going to have short stature, obesity, hyperphagia, low impulse control, hypotonia with hypopigmentation. All this is the Prader-Willi syndrome. Prader-Willi syndrome is because of what? Prader-Willi syndrome is because of the deletion of chromosome number 15. Paternal deletion. Now, what are the important points about the Angelman syndrome? Now look at the Angelman syndrome. So this Angelman syndrome, these babies, for example, this female, now she had Angelman syndrome. Now whenever you look at this Angelman babies, they are going to have this uncontrollable laughter. Okay, they are going to have this weird smiling. Okay, weird smiling. So these Angelman babies, they are called as happy puppets. Okay, they are called as happy puppets, sir. They are called as happy puppets. They are having this uncontrollable laughter, sir. Okay, that's the one important point which I want you to know. See, they are having this weird smile. Okay, weird smile is going to be there. And one more important point is these patients are going to have the main problem for these patients is seizures. Angelman syndrome. So seizures are going to be there, uncontrollable laughter is going to be there, and these patients are usually mentally retarded. Okay, mentally retarded, sir, intellectual disability is going to be seen. So image based question, this is the Prader-Willi boy, okay, and this girl is having that uncontrollable laughter, she is having Angelman syndrome. So just a recap, sir, SNRP genes are active. Remember, the SNRP genes are active in father. There is maternal silencing. And how do you do silencing? MCQ. Silencing is done by doing methylation and UBE3A genes. Okay, UBE3A genes. So the UBE3A genes, they are silenced in the father by doing methylation. UBE3A genes are active only in mother. Okay, so this is done, sir. We have also completed the topic of genomic imprinting. So now completed the important non-Mendelian disorders, the non-Mendelian inheritance, completed, sir. Now after this, guys, can you tell me this image based question, what exactly are we discussing? Now by looking at this image, can you tell me, sir, this baby, this female, is she suffering with which condition? Can you tell me? Sir, actually now we are going to discuss about the chromosomal disorders. Means now we are not talking about the genes, sir. We are not going to talk about the genes. Entire chromosomes, the chromosome is deleted, or having an extra chromosome. We are not talking about the single genes. We are not talking about the point mutations. We are talking about the entire chromosomal problems. Chromosome got deleted, chromosome have inverted, translocations, deletions, insertions. We are going to discuss about them. Okay. Now, so the first disorder which I want to discuss is Down syndrome, sir. Okay, Down syndrome. Now, my questions to you. So this Down syndrome is because of which chromosomal abnormality? What is the chromosomal abnormality seen in the Down syndrome? It's a trisomy. Trisomy what? Trisomy 21. Trisomy 21 means there is an extra 21 chromosome. Okay, there is extra 21 chromosome. Normally, in your cells, how many 21 chromosomes are supposed to be there? Sir, there are supposed to be 2 chromosomes, 21 chromosomes, one from the mother, one from the father. But if you get one extra 21 chromosome, sir, then it is going to cause Down syndrome. That's the trisomy 21, extra 21 chromosome. Now, what are the causes? Why Down syndrome? Why? So the most important risk factor, let me write here. Risk factor is increase in maternal age. Increase in the maternal age, which means now this female is 35 years or 37 years. Now at this age, if she is trying to conceive, if she is trying to have a baby, that baby might develop, or that baby might get this Down syndrome, sir. Down syndrome. So the causes, important cause is the non-disjunction. Okay, meiotic non-disjunction, sir. Okay, meiotic non-disjunction in 95% of the cases. During meiosis, normally the chromosomes are supposed to separate, right? The chromosomes are supposed to separate. Okay, during meiosis, the chromosomes number should cut down into half, should become half. Okay, the 46 chromosomes should split, and it should, like in one cell 23 chromosomes, and into the other cell 23 chromosomes. It should be segregated, but non-disjunction, this 21 chromosome is not separating. Okay, so in one single cell, let me put it this way for your understanding. Sir, now there is this ovum. Okay, ova. Now in this ova, there are 221 chromosomes. 221 chromosomes. Sir, mother is giving the ova, and in this ova, there are 221 chromosomes. Why? Because the chromosomes are not separated, not split into two halves. Actually, there should be only 121 chromosome, but here there are 221 chromosomes. Now such an ovum, if it is fertilized by a sperm, sir, this is the sperm, sir. Now even sperm have 121 chromosome. Now tell me what happens. Sir, here 2 chromosomes. Now here 121 chromosome. Now what happens in the zygote? Sir, in the zygote there will be, in the baby there will be 321 chromosomes. Okay, that's a non-disjunction. Non-disjunction. Maternal non-disjunction, sir. Maternal. So mother age is increasing. So her meiosis, like her oocytes, the quality of the oocytes she is producing, they will gradually decrease. The quality decreases. There will be more and more non-disjunctions that are happening. Because of this non-disjunction, there can be Down syndrome. 95% of the cases there is a maternal non-disjunction during meiosis. Other causes, other cases includes, okay, Robertsonian, translocation, which is accounting for 4% of the cases, Robertsonian translocation, or mitotic non-disjunction. Mitotic non-disjunction can also lead to Down syndrome, but only in very few conditions, or very few conditions, 1% of the cases. Now you need to know the most important for your FMG exams, the most important point, and for the NEET PG exam also, is the non-disjunction of maternal 21 chromosome during meiosis, accounting for 95% of the cases. Okay, that's the important point I want you to know. So after this, sir, how the Down's babies are going to be? If you look at the Down's babies, what are the clinical features that are seen? Now look here, this is the Down's baby. Now what you can appreciate is a big tongue that is protruding out. Okay, that's a macroglossia you can see. Now if you look at their the babies, if you look at their legs, you can see a very much gap between the toe and the other fingers. This is called as saddle toes. Okay, saddle toes. And if you look at their eyes, you can see in the iris there are the spots seen. These are called as brush field spots. Next, if you look at their hands, there will be one single line. This is called as palmar simian crease. Okay, palmar simian crease is going to be seen. Now let's try the clinical features, sir, one by one. One by one, clinical features. The clinical features are going to be, first clinical feature number 1, there will be mental retardation. Now not only this, sir, remember, for your entire life, remember, Downs, Edwards, Patau, all the chromosomal disorders are going to have the mental retardation, no doubt. Not just the Downs, but also Edwards, Patau, Cry-Ducat, all the chromosomal disorders are going to present with the mental retardation. And this Downs is the most common cause of mental retardation. Okay, Downs is the most common cause of mental retardation in the chromosomal disorders. Okay, most common cause of mental retardation. Next, these patients are going to have macro. We have seen glossia. Okay guys, macroglossia, the big tongue is going to be seen. In the eyes, what you have seen? It's the brush field spots. Next, palmar simian crease. Okay, palmar simian crease. Okay, palmar simian crease is going to be seen. Next, what else? If you look at their eyes, okay, brush field spots are seen, but if you look at their eyes, so there will be upslanting palpebral fissures. Okay, their eyes will be slanting like this. Okay, so if you look at their eyes, they are little like this. Okay, there will be upslanting. Palpebral fissures. And if you look at their nose, the nose is going to be flat. Okay, the nasal bridge, the nasal bridge is going to be flat, sir. Flat nasal bridge. Okay, the nasal bridge is going to be flat. And what else we have missed? In the toes, saddle toe. Saddle toe is going to be seen. They are going to have the saddle toe. So macroglossia, brush field spots, palmar simian crease, oblique palpebral fissures, flat nasal bridge, and they are going to have the saddle toes. And you can say, like, we can put many more points, like, their faces are going to be like mongoloid faces. Okay, mongoloid faces, like, ape-like, okay, mongoloid faces. Okay, so all these are the important points which I want you to know regarding the Down syndrome. Apart from this, so if I am having Down syndrome, okay, the external features I have explained. Internally, sir, which diseases I can get? What are the problems with the cardiovascular system? What are the problems with the blood? What are the problems with the, like, different organ systems, kidneys and GIT? Now, diseases associated with Downs. See, these Downs patients, by age of 40 years, by the time they reach their 40 years, okay, 30-40 years, some books mentions 30 and some books mentions 40, by the time they reach 40 years, all these Downs patients, they are going to develop Alzheimer's disease. That's the dementia, sir. They will develop this Alzheimer's disease. This is the one important point I want to know. In cardiovascular system, okay, what is the cardiovascular problem? So these patients are going to have congenital anomalies. The serious congenital anomalies, endocardial cushion defects. Endocardial cushion defects. Endocardial cushion defects are going to be seen. Next, GIT abnormalities. These patients will have its sprung disease. That's a congenital megacolon, Hirschsprung disease. And in GIT, these Downs babies, they will develop the duodenal atresia. Okay, duodenal atresia, sir. So this is a very important MCQ. The Down syndrome patients are going to develop the duodenal atresia and radiological sign. On radiology, how the duodenal atresia is going to look like? Double bubble appearance. Double bubble appearance, sir. Okay, and we have completed the Alzheimer's disease, endocardial cushion defects are completed, GIT abnormalities completed. Now, hematological abnormalities. See, the point which I want you to know is the most common, if you ask me, the most common hematological abnormality. Do you know? Sir, what is the most common hematological abnormality? The most common hematological abnormality is seen in the Downs patients. Do you have any idea, guys, what is the most common hematological abnormality that is seen? It's a transient, see, it's not a cancer, sir. It's a transient abnormal myelopoiesis. Transient abnormal myelopoiesis. Transient abnormal myelopoiesis is the most common hematological abnormality that is seen. But if they ask you, okay, in Down syndrome, what are the cancers? Okay, the blood cancers, which blood cancers are most common? Before 3 years, okay, this is very important. Before 3 years of age, what is the most common cancer that they will develop? It is acute megakaryoblast leukemia. Okay, acute megakaryoblast leukemia. Okay, acute megakaryoblast leukemia. After 5 years, which cancer they can develop? Okay, which blood cancer they can develop is Alleles, acute lymphoblastic leukemia. Okay, ALL, acute lymphoblastic leukemia after 5 years of age. Before 3 years of age, the most common cancer that they can develop is acute megakaryoblast leukemia. Okay, acute megakaryoblast leukemia, sir. These are the problems which are seen, these are the diseases which are associated with the Down syndrome. Okay, Alzheimer's disease, endocardial cushion defects, Hirschsprung disease, duodenal atresia. Most common hematological abnormality is a transient abnormal myelopoiesis. Before 3 years, it is acute megakaryoblast leukemia. After 5 years, it is acute lymphoblastic leukemia, sir. Now, in your exams, what else they can ask you is, देखो, screening tests. Okay, screening test. Now imagine there is this one female. Now she is trying to conceive, she is trying to have a baby at 38 weeks, at 38 years. Okay, now she is 38 years and now she is trying to have a baby. Now increase in maternal age is risk factor for development of the Down syndrome. You know it. Now, you want to know whether the baby, okay, that she is having, the baby is suffering with Downs or not. You just want to know it. So what you will do? So you are going to order for ultrasonography, right? Ultrasonography. So in ultrasonography, what you can see, okay, in ultrasonography, okay, actually I just kept an image. I forgot. Okay, I think it was deleted. So in ultrasonography, what you can see is there is increase in nuchal translucency. Okay, increase in nuchal translucency, very important MCQ. So the nuchal translucency, in the nuchal area, in the neck area, there will be a very big gap. Nuchal translucency, it will be like black in color. So increase in nuchal translucency is associated with Down syndrome, sir. On ultrasonography, whenever you see the increase in nuchal translucency, that indicates that the baby is suffering with the Down syndrome. Apart from this, apart from ultrasonography, how can you do like some other confirmatory tests? The other confirmatory tests includes, now there is a test called as dual testing. Dual testing. Now in this dual testing, you are going to check for two markers. One is beta HCG and PAP-A. Okay, pregnancy associated plasma protein A. So when you do this dual testing, okay, especially in the first trimester, when you do this testing in the first trimester, now the beta HCG levels in the mother are very much very much elevated, very highly elevated, and PAP-A levels are going to be decreased. Whenever you do dual testing which shows high levels of beta HCG and low levels of PAP-A in the mother, now it is indicating, it is giving you a signal that the baby is having Down syndrome. The baby might be suffering with the Down syndrome. In second trimester, you can do one more test which is called as a triple test. Triple testing. Now in this triple testing, what are the markers you are going to check for? The markers you will be checking for is again beta HCG. Also you will be checking for alpha fetoprotein and estrogens. Okay, estriol. Estriol is a major estrogen during the pregnancy. Estriol, sir. Now what happens? Sir, beta HCG levels are elevated, alpha fetoprotein and estriol levels are reduced. Now whenever the alpha fetoprotein levels and estriol levels are reduced with elevated beta HCG levels, then it's indicating that the baby is suffering with Down syndrome. Now there is one more test which is called as a quadruple test. Quadruple testing. In quadruple testing, what you will check? Beta HCG, the markers, the beta HCG, alpha fetoprotein, same estriol. Along with estriol, you are also checking inhibin levels. Inhibin A levels. Now beta HCG levels are elevated, inhibin A levels are elevated, but alpha fetoprotein levels and estriol levels are less. So this, like in quadruple testing, this was the question asked in 2019 June exam. So in quadruple testing, along with beta HCG, alpha fetoprotein and estriol, what else? What is the fourth marker? The fourth marker is inhibin A. So by doing these three tests, these are the screening tests, dual testing, triple testing as well as quadruple testing. These are the screening tests by which you can say that the baby is suffering with Down syndrome. Okay, now done, sir. After Down syndrome, now the other chromosomal disorder that I am showing you here. Guys, can you tell me what is this chromosomal disorder? Where, see, look here, the baby is palate. The palate is having a problem. There is a cleft palate. Okay, you can very clearly see here, there is a cleft palate, sir. And you can see polydactyly. And there is a hole here in the iris. It's looking like a keyhole. Now it's looking like a keyhole. What is this condition called as? This condition which I am showing you, this is called as Patau. Okay, Patau, I used to remember Patau for palate, P for P. Okay, Patau, sir. Patau syndrome. So what is the problem in Patau syndrome? So it is trisomy 13. Okay, trisomy 13, extra 13 chromosome. Okay, chromosome number 13 is extra, which means a cell is having 13 chromosomes, 13. And I have said you, all chromosomal disorders are going to have mental retardation. So even this Patau syndrome, the patients are going to be mentally retarded, mental retardation, trisomy 13. Now, for your exam purpose, what else you should know? Dekho, right? There is cleft lip and cleft palate. Cleft lip and cleft palate is going to be seen. See here, one more P. Okay, that is polydactyly. Polydactyly, extra fingers, polydactyly is going to be seen, sir. Now, these patients are going to have a hole in the iris that is called as keyhole coloboma. Keyhole coloboma, sir. Okay, so you can very clearly see it just looking like a keyhole. You put the key, right? Keyhole, sir. So Patau syndrome, the patients are going to have the mental retardation. Polydactyly is going to be seen. Cleft lip and palate is going to be seen. Keyhole coloboma. And if you want to know, these patients are also going to have patent MCQ, patent ductus arteriosus. And these patients are also going to have the rocker bottom feet. Yeah, most of the students will think rocker bottom feet is seen in the Edwards syndrome. Not only in Edwards syndrome, rocker bottom feet is also seen in Patau syndrome. So these are the important points I want you to know regarding Patau. Okay, if you know this much, enough, sir. Now after this, the next, look here. Guys, can you tell me? Here the patient, here the boy is having, see, dekho, now is a rounded feet. The feet is now rounded, and you can see the skull, it is in the shape of a strawberry. It's in the shape of a strawberry. So what is this syndrome which we are discussing here? This is called as Edwards syndrome. Edwards syndrome. Now in Edwards syndrome, what's the problem? Again trisomy. Trisomy what? Edward 18, trisomy 18. So 3 extra chromosomes. 18 chromosomes. Okay, 1 extra 18 chromosome. So these patients are going to have again mental retardation. Okay, now they are going to have, like I said you, it's a strawberry skull. Okay, strawberry shaped skull. Why it is strawberry shaped? Because see, they are going to have small head with a prominent occiput. The occiput is going to be more prominent and the small head, sir. So this strawberry shape, it is because of microcephaly. Microcephaly, small head with a prominent occiput. Okay, microcephaly with prominent occiput is going to be seen. Apart from this, what else are the clinical features which you should know regarding the Edwards syndrome? So these patients are going to have a kidney abnormality. Very important MCQ. Sir, this Edwards syndrome is associated with horseshoe kidney, sir. Okay, horseshoe kidney is going to be seen. And if you look at their feet, see, what is this? This is called as the rocker bottom feet. Rounded feet, sir. Okay, rocker bottom feet are going to be seen. Okay, rocker bottom feet are going to be seen. And if you look at their hands are going to be like this, clenched hands. Okay, clenched hands. Clenched hands or clenched fingers. So trisomy 18, important point. What are the important points which you need to know, guys? It's the strawberry skull, microcephaly, prominent occiput with the rocker bottom feet, and clenched hands. Horseshoe kidney is going to be seen. So these are the important points about the Edwards syndrome. What we have completed? We have completed the Down syndrome. See, regarding Down syndrome, just as a recap, Down syndrome, what are the important points? Trisomy 21, mental retardation, okay, the mongoloid faces, upslanting palpebral fissures, okay, oblique upslanting palpebral fissures are going to be seen, okay. Next, mongoloid faces, palmar simian crease, brushfield spots are going to be seen. Now these Downs patients are going to develop Alzheimer's disease by the age of 40. Downs patients are associated with endocardial cushion defects, Hirschsprung disease, okay, duodenal atresia is going to be seen. Most common hematological abnormality is abnormal Myelopoiesis. Okay, abnormal myelopoiesis is going to be the most common hematological abnormality, but before 3 years it is acute megakaryoblast leukemia, but after 5 years it is ALL. Downs patients, important points completed. Now after that, Patau. What are the important points about the Patau? So Patau, the patients are going to have the cleft lip and palate. Polydactyly is going to be seen. Okay, keyhole coloboma is going to be seen. Okay, rocker bottom feet is going to be seen. Okay, done. Enough. After this, we have discussed about the Edwards syndrome. Edwards syndrome, trisomy 18, mental retardation, strawberry skull, prominent occiput, microcephaly, rocker bottom feet, horseshoe kidneys. Done, sir. Enough. After this, let's discuss about now Turner syndrome. Turner syndrome. Sir, what is the problem with the Turner syndrome? See, the problem, the chromosome, again chromosomal abnormality, sir. But here, it's not the extra chromosome. One chromosome is lost. What is that? What is that chromosome that is lost? It's the monosomy X, which means normally a female was supposed to have 46XX, right? 46XX. A female is supposed to have 2 X chromosomes. Now one X chromosome is gone. X0, 45X0 or XO. One chromosome is lost. One X chromosome is lost. So it is monosomy X, sir. Okay, it's monosomy X. Okay, and most of the time the missing X, the chromosome which is lost, that X chromosome which is lost is paternal in origin. So the paternal X chromosome is lost, sir. Okay, see, this Turner syndrome, there is no increased risk with increased maternal age. So with the increase in maternal age, there is a risk of Edward, there is a risk of Downs, there is a risk of Patau, but there is no increased risk of Turner syndrome with increased maternal age. Okay, now this is the image based question which was asked many times, in the exam. See, dekho, now there was supposed to be 2 X chromosomes. There was supposed to be 2 X chromosomes, but now here X chromosome is gone. There is only 1 X chromosome. Dekho, there is only 1 X chromosome. Now, what are the clinical features? If a female is not having 1 X chromosome, what are the clinical features? I used to remember, like, if 2 X's are present, she is a complete female. If she is having only 1 X, she is the half female. Everything is going to be half. She is the half baked female now. Okay, just like a half baked cake. Now she is a half of 50% female. Now what are the problems? So she is going to have a webbing of the neck. Okay, webbed neck. She is going to have low posterior hairline. Now she is going to have a shield shaped chest. Okay, she is not going to have a proper breast. Her chest will look like a shield, sir. Okay, widely spaced nipples with shield shaped chest. The nipples are going to be widely spaced because her chest is like a shield. So the nipples are widely spaced. Next, if you look at her hand, the fourth metacarpal is going to be short. Okay, the fourth metacarpal is going to be sharp, and she is going to have the cubitus valgus. She is going to have hands something like this. Okay, cubitus valgus. I will show you the image. She is going to have the cubitus valgus, sharp, short fourth metacarpal, low neck hairline with a webbing of the neck, widely spaced nipples, and shield-shaped chest. Now, MCQs. What are the serious anomalies that are seen? The most common serious anomalies are the two. Two are there. One is the bicuspid aortic valve. Okay, bicuspid aortic valve and coarctation of aorta. Okay, these are the two serious anomalies that are seen. Bicuspid aortic valve is associated with the Turner's, true, and coarctation of aorta. Renal anomalies. Turner females can develop horseshoe kidney, true. Now, just now we have seen, sir, horseshoe kidney. It is seen with what? Edwards, or it is seen with Patau? Sir, look here. It is seen in Edwards, sir. Edwards syndrome patients will have horseshoe kidney, true. Even Turner female can have horseshoe kidney. Now, these Turner females, they are at a risk of, increased risk of autoimmune diseases. Okay, all autoimmune diseases are most commonly seen in females. Now, even this Turner female, she is at a risk of developing diabetes and Hashimoto's thyroiditis. Okay, diabetes and Hashimoto's thyroiditis, these are the autoimmune disorders, true. But very, very important point is her IQ is going to be normal. Okay, it's not a mental, there is no mental retardation. Okay, in trisomies, in all the trisomies, there is mental retardation. But here, in Down syndrome, very, very important, her IQ is normal, sir. She is just like a normal regular girl, but she is short, shield chest, widely spaced nipples, webbing of the neck, low posterior hairline are seen. But IQ is normal. If you look at her ovaries, okay, if you look at her ovaries, the ovaries are totally like not properly formed. It's a streaky ovaries. The thin sliver of ovarian tissue is present. Thin sliver of ovarian tissues. So there are streaky ovaries. Streaky ovaries. Thin sliver of ovarian tissue is present. So is she going to have the proper menses? No, she is not going to have the proper menses, and she will be infertile. Okay, these females are going to be infertile in majority of the cases. In majority of the cases, these are infertile. Why? Because the ovaries are not properly developed. Okay. So they are going to have the less estrogen levels. Ovaries are not properly there, so they are going to have less estrogen levels. So what happened to FSH and LH? They are elevated. FSH and LH are elevated, but the estrogen levels are less. Less amount of estrogen levels are seen. Okay. Now the same things I have discussed every clinical feature. Now you can very clearly see, sir, this female, this is a normal female which I am showing on the one side. So this female, see, she is having coarctation of aorta, bicuspid aortic wall, cubitus valgus, her hands are going to be something like this, short fourth metacarpal, streaky ovaries, autoimmune diseases like diabetes mellitus, horseshoe kidney, shield-shaped chest, widely spaced nipples, webbing of the neck, and low posterior hairline. Each and every point I have discussed, sir. Every point is discussed. Okay. Now, look here. See this Turner's. One important point. Turner's is the most common cause of what? Turner's is the most common cause of primary amenorrhea. Okay. See, if there is this one female who is suffering with the Turner's, she is not going to get her menses. By the age 12, 13, a normal female will get her menses, but this Turner female, she is not having her ovaries properly, not developed. So do you think is she going to have the normal estrogen? No estrogen. Is she going to have the ovulation? No ovulation, no progesterone, so no menses, sir. So it's the most common cause of primary amenorrhea. Okay. So problem is gonadal dysgenesis. Ovaries are not formed properly. So gonads are not formed properly. There is a dysgenesis, Turner syndrome. Okay. So this Turner syndrome, in Turner syndrome, one more important point I want you to know, this was a question asked. Sir, there is hypergonadotropic. Hypergonadotropic means the gonadotropic hormones. There is no problem in the brain. There is no problem in the brain. There is no problem in the hypothalamus, or there is no problem in the anterior pituitary. So FSH levels are elevated. LH levels are elevated. There is high FSH, high LH, sir. Why? Because ovaries are not developed. When the ovaries are not developed, there is no estrogens. Estrogens are not there. Progesterone is not there. When estrogen is not there, there is no negative feedback. When progesterone is not there, there is no negative feedback. As there is no negative feedback, the FSH levels are elevated, LH levels are high, but estrogen levels and progesterone levels are low. So that's why hypergonadotropic. Gonadotropic hormones are more, FSH is more, LH is more, but hypogonadism. Gonads are not there, ovaries are not there, hypogonadism. So hypergonadotropic, hypogonadism. Example is a Turner's. Okay. So all important points about the Turner's are completed, sir. The last chromosomal disorder which I want to discuss here is Klinefelter. What is the problem with the Klinefelter? Very simple thing. Sir, it's going to occur in the males. Why? What is the problem? So 47XXY, the most common, the genotype abnormality is 47XXY, which means one extra X chromosome. Normally it should be 46XY, right? A male is supposed to have, for example, 46X and Y, one X chromosome, one Y chromosome. If there is an extra X, now it becomes 47 chromosomes are there. What is that extra chromosome? One extra X chromosome is there. For example, this is, for example, 48XXXY. This is also Klinefelter. This is also Klinefelter, no doubt. So extra X chromosome is simple Klinefelter. Okay. Most common genotype is 47XXY. Now what happens? Normally XY is male, XX is female. Now if a male is having one extra X, X is more female, right? If a male is having one extra X, female-like characters will develop. Female-like characters. What are those female-like characters? Now gynecomastia, dekho, now he is having breast tissue development. Gynecomastia. True. Now if you look at his testes, the testicular atrophy will occur. There is testicular atrophy. Okay, there will be micropenis, testicular atrophy, and even this testis, there are no sperms. Azoospermia, MCQ, there are no sperms. But if you look at their height, they are going to be tall statures. So that their height is going to be tall. They are tall stature with gynecomastia, azoospermia, and if you talk with them, the voice is going to be more girlish. Okay, that's high pitch voice. high pitch voice. Okay, they are going to have the high pitch voice. And are they going to be fertile now? No, they are not going to be fertile because there are no sperms at all. Okay, they are going to suffer with infertility. And yeah, in some patients there will be mental retardation. Some patients with Klinefelters will be normal also. There are variations are there. Okay, but anyway, Klinefelters is a male who is almost having the feminine characters like high pitch voice, gynecomastia. Okay, gynecomastia. And if you look for the sperms, there are no sperms. As there are no sperms, there is infertility. Okay, there is infertility, sir. So now these patients, they are at a risk of breast cancer. Also, this is also important point which I want you to know. They are at risk of breast cancer. So the Klinefelters. So Klinefelter is an extra X chromosome. So with this, all the chromosomal disorders also I have completed. The Turner-Edwards-Patau, Turner syndrome as well as the Klinefelter syndrome. So with this, all the important points, whatever you need to know for your exams are completed.